Assalamualaikum warahmatullahi wabarakatuh. All praise and gratitude be to Allah, the Lord of the world. May peace and blessings be upon our Prophet Muhammad sallallahu alaihi wasallam.
The Honorable Rector of Universitas Syiah Kuala, Profesor Dr. Insinyur Marwan.
The respected Chairman of Konsorsium Biologi Indonesia, Profesor Dr. Budi Setiadi Daryono.
The respected Dean of the Faculty of Mathematics and Natural Sciences, Universitas Syiah Kuala, Profesor Dr. Taufik Fuadi Abidin.
The respected Dean of the Faculty of Teacher Training and Education, Universitas Syiah Kuala, Dr. Syamsur Rizal.
The respected Dean of the Faculty of Science and Technology, Universitas Islam Negeri Ar-Raniri Banda Aceh, Dr. Insinyur Muhammad Dirhamsyah.
The respected Head of Biology Department of the Faculty of Mathematics and Natural Sciences, Universitas Syiah Kuala, Profesor Dr. Leni Fitri.
The respected Head of Biology Education, Department of the Faculty of Teacher Training and Education, Universitas Syiah Kuala, Dr. Muhib Budin.
The respected Head of Biology Department of the Faculty of Science and Technology, Universitas Islam Negeri Ar-Raniri Banda Aceh, Dr. Musli Hidayat.
The respected Chairperson of the International Conference on Biology and Environmental Sciences 2024, Mrs. Essi Arnelli, PhD.
The respected keynote speakers, Profesor Dr. Iskandar Zat Siregar, Profesor Dr. Suhartono, and Association Profesor Dr. Muhammad Izzuddin Syakir Ishak.
The respected invited speaker, Mr. Rudi Hadian Syahputra, Dr. Linsai F. Banin, Mr. Cijenko, and Dr. Marius Ekwe.
The respected moderators, Dr. Insinyur Wira Dharma, Mrs. Elizar PhD, and Mrs. Zaudatul Ulia.
The respected affiliates who have contributed to success of this event.
And to all the respected attendees for today's international conference. Welcome to the International Conference on Biology and Environmental Sciences 2024.
By reciting Bismillahirrahmanirrahim,
the opening ceremony of International Conference on Biology and Environmental Sciences, Wednesday, November 6, 2024, has now begun.
To bless our event today, let us listen to recitation of the Holy Quran.
We would like to invite Irfan S. to the stage.
Assalamualaikum warahmatullahi wabarakatuh. أعوذ بالله من الشيطان الرجيم Bismillahirrahmanirrahim.
يا أيها الذين آمنوا اتقوا الله حق تقاته ولا تموتن إلا وأنتم مسلمون واعتصموا بحبل الله جميعا ولا تفرقوا واذكروا نعمة الله عليكم إذ كنتم أعداء فألف بين قلوبكم فألف بين قلوبكم فأصبحتم بنعمته إخوانا وكنتم على شفا حفرة من النار فأنقذكم منها كذلك يبين الله لكم آياته لعلكم تهتدون ولتكن منكم أمة بالخير ويأمرون بالمعروف وينهون عن المنكر وأولئك هم المفلحون وأولئك هم المفلحون صدق الله العظيم.
Assalamualaikum warahmatullahi wabarakatuh.
Thank you to Irfan S.
Next, we will sing the Indonesia Raya anthem and the Achinese hymn. We would like to invite Anna Maria to the stage, and we kindly ask the audiences to stand.
Siga membela bangsa, mulia nanggro mulia dum syuhada, metuah bijeh Aceh mulia. Ya Tuhanku, rahmat bersampo, negeri Acehnya bermulia. sepakat bersabarnya menemat syariat Islam ke hukum bangsa. Ya Tuhanku, rahmat bersampo, negeri Acehnya bermulia. Rahmat melimpah, metuah asa, areh kekamo bersejahtera. Aceh masyhur makmungan megah, sabeta jaga Aceh mulia. Aceh masyhur makmungan megah, sabeta jaga Aceh mulia.
All audiences may now take their seats again. Thank you to Ana Maria.
Next, the report from the chairperson of ICBIS 2024. We would like to invite Mrs. Essie Harnelli, PhD, to the stage.
Mrs. Ceci Arnelli, PhD, is a biology lecturer at the Faculty of Mathematics and Natural Sciences, Universitas Syiah, Kuala, specializing in plant molecular biology, climate change related to plant biodiversity, and DNA barcoding.
Assalamualaikum. Udah. Hah? Pegang aja. Assalamualaikum warahmatullahi wabarakatuh. First of all, let us express our gratitude to Allah Subhanahu wa Ta'ala for His mercies and blessing that have allowed us to gather here in good health. May salawat and salam be upon our Prophet Muhammad sallallahu alaihi wasallam. Excellencies, distinguished delegates, esteemed guests, speaker, participants, ladies and gentlemen, as a chairwoman, it is my great honor to welcome you all to the first International Conference on Biology and Environmental Sciences, ICBIS, hosted by the Biology Department, Faculty of Mathematics and Natural Science, reflecting the theme of aligning biodiversity and climate initiative for a sustainable future. We are happy to gather scholars, researchers, and practitioners from around the globe to exchange ideas, share knowledge, and foster collaborations with the vital fields of biology and environmental sciences. We would like to express our gratitude to Prof. Budi Setiadi Daryono and Kobi for encouraging us to conduct the first international conference from Biology Department, Faculty of Mathematics and Natural Science, Universitas Syiah, Kuala. Hopefully, we can continue to organize this kind of seminar regularly in the future. Our gratitude for distinguished keynote speakers, Profesor Dr. Iskandar Siregar from IPB University, Indonesia, Profesor Dr. Suhartono SSIMS from Universitas Syiah Kuala, Indonesia. Associate Professor Dr. Muhammad Izzuddin Syakir Ishak from University Science Malaysia. I would like also to express my gratitude to our invited speakers, Marius Ekwe from Alliance Biodiversity International Cameroon, Dr. Chijenko from the Global Biodiversity Information Facility, GBIF, Regional Asia, Taiwan, Dr. Lindsay F. Banin from UK Center for Ecology and Hydrology, United Kingdom, and Rudi Hadian Saputra, MSc from Loser Conservation Forum, Indonesia. Your expertise and insight will enrich our discussion and inspire new direction in our research and practices. Furthermore, your involvement improves the quality of our conference and emphasize the relevance of international collaboration in addressing challenges in our area of study. The conference used a double-blind review process for all submitted papers, receiving more than 50 abstracts. After thorough evaluation, 50 abstracts were accepted for presentation, with 32 falling within the basic biology field, 13 in interdisciplinary biology, and 5 in environmental sciences. This paper from participants connected with Indonesian foreign university, companies, or institutions are eligible for publication in our Scopus and Sinta Index partner publication. Our participating university included Universitas Syiah Kuala, Universitas Cendrawasih, National Research and Innovation Agency, BRIN, Universitas Islam Malang, Sumatran Institute of Technology, Universitas Bengkulu, Universitas Kristen Jakarta, Universitas Islam Negeri Araniri, Bandung Institute of Technology, University Science Malaysia, Alliance of Biodiversity International Cameroon, Ain Shams University Egypt, Süleyman Demirel University Turkey. We are grateful to the outstanding person who contribute to the success of this meeting. First and foremost, we would like to express our sincere thanks to the Rector of Universitas Syiah Kuala, Prof. Dr. Insinyur Marwan. Thank you, Prof. Marwan, for supporting us with the incentive for the international conference. A profound thanks to our Dean, Professor Dr. Taufik Fuadi Abidin SSI MTech, and Professor Dr. Mudatsir, the head of the Institute for Research and Community Service at Universitas Syiah Kuala. Finally, we would like to thank all the authors who submitted their papers, the reviewers, the keynotes, speakers and invited speakers, the moderators, the committees, and our volunteers for their works preparing and running the conference. We wish you a very warm welcome and hope that you will have a very productive and enjoyable time. Once again, I'm most grateful for your participation and support. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Genetics and breeding.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. The Honorable Rector of University Syiah Kuala, Professor Marwan. The Honorable the Dean, Faculty of Mathematics and Natural Sciences, Professor Taufik, and also along with the Dean of Faculty Teaching and Education Sciences, as well as also the Dean of Faculty of Science and Technology, Islamic State University, Araniri. Also distinguished presenters and guests, ladies and gentlemen. Good morning. We convey Alhamdulillah, all the praise to Allah Subhanahu wa Ta'ala, because yesterday, by blessing the Allah Subhanahu wa Ta'ala, we finish and complete our Kobe National Meeting. And on behalf of the Kobe, we would like to warmly welcome you to the International Conference on Biology and Environmental Sciences, ICB's 2024, with the term Aligning Biodiversity and Climate Initiative for a Sustainable Future. Congratulations again to all the committees. This term has been selected to carry on useful idea for better human life and environmental sustainable development. Since the establishment of the first National Congress, Kobe, in 2016 in Uin Alauddin, Makassar, so the Kobe always support and collaborated with many universities in Indonesia for conducting some international conference, such as today, ISBs, which is organized by the Department of Biology, Faculty of Mathematics and Natural Sciences, in collaboration with the Department of Educational Biology, Faculty of Teaching and Education Sciences, Universitas Syiah Kuala, and also along with the Department of Biology, Faculty of Science and Technology, UIN Araniri. The ICBs will serve as a platform for researchers and scientists. The most important is to meet and share the most recent progress in the field of biology and climate initiative. I do believe that nothing is more important to advising biodiversity and climate initiative in this area than ensuring a better networking and sharing new findings among national and international researchers and scientists. On behalf of the Kobe, we would like to thank all keynote and invited speakers for sharing research results and new findings in the ISBs. And we are indebted to organizing committee led by Dr. Eli Harnelli, along with Professor Lenny yesterday and friends, for working hard to make the ISBs a success and come to reality. Finally, I'm sure that the conference will have a fruitful exchange for idea on biodiversity and climate initiative and bring the audience to exchange and brainstorm the scientific knowledge. And of course, to make this earth a better place for all living beings. Finally, before I end my speech, I would like to convey a very short rhyme. I see the eagle, and then I took a picture. The international is a struggle, but to achieve your future. Thank you so much. Enjoy and have a great scientific meeting. Good morning. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you to Professor Dr. Budi Setiadi Daryono.
Next, welcoming speech by the Dean of Faculty of Mathematics and Natural Sciences, Universitas Syiah, Kuala. We would like to invite Professor Dr. Taufik Fuadi Abidin to the stage.
Professor Dr. Taufik Fuadi Abidin is the Dean of the Faculty of Mathematics and Natural Sciences, Universitas Syiah, Kuala, and a professor in the field of computer science.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Hamdan wa syukurillah, salatan wa salaman ala rasulillah. Distinguished Rector Universitas Syiah Kuala, Profesor Dr. Insinyur Marwan, thank you very much for your time, Bapak Rektor. Distinguished Head of KOBI Consortium, Professor Budi Setiadi, thank you very much. Distinguished guests, the head of the Institute for Research and Community Service, Professor Mudasir, Vice Dean, distinguished guests, honorable speakers, and all participants. Good morning and welcome to the Faculty of Mathematics and Natural Sciences, shortened as FMIPA, Universitas Syiah Kuala, for the first international conference on biology and environmental science, or shortened as ICBES. This first conference is organized by the Department of Biology at our faculty. It is a privilege for our faculty to host this important event. As a faculty dedicated to advancing scientific knowledge and achieving excellence, we strive to become a center of excellence in education and research in the field of biology, environmental science, and beyond. FMIPA is committed to producing impactful research and innovation that addresses critical issues, particularly in environmental sustainability, biodiversity conservation, and climate change. Through active collaboration and interdisciplinary approach, such as this first international conference, we aim to make meaningful contribution not only to Indonesia, but also to the global scientific community. Rector of Universitas Syiah Kuala, Prof. Budi, and distinguished guests, as one of the leading faculty at USK, FMIPA is recognized for its academic and research achievement, such as in the field of biology. We have so many professors, doctors, scientists, and also in the environmental science. Inshallah, Pak Rektor, next year and in the following years, we are planning to open the undergraduate program in environmental science. Hopefully, we will have support, full support from Bapak Rektor. Our state-of-the-art facilities, combined with highly skilled faculty members, allow us to conduct cutting-edge research that inform policies and provide a real-world solution. We are proud to support this academic environment in which students and researchers and also faculty members encouraged to explore, innovate, and advance scientific understanding. I hope this conference will become a great forum for professors, for scientists, practitioners, policymakers, and also students to discuss, exchange ideas, and develop tangible strategies to address environmental issues. We believe that our faculty plays an essential role in promoting interdisciplinary research, innovation, and contributing to global.
Thank you to Professor Dr. Taufik Fuadi Abidin.
Next, welcoming speech and opening by Rektor Universitas Syiah Kuala. We would like to invite Professor Dr. Insinyur Marwan.
Profesor Dr. Insinyur
Honorable Chairman of Consortium of Biology Indonesia, Vice Dean, Head of Research Institute, Universitas Syiah Kuala, the committee of the first time hosting this very important meeting, for your support, encouraging, and motivation, so our colleague from the Department of Biology can start the first ICBS. It is an honor and privilege for Universitas Syiah Kuala to host this very important conference. As a university committed to achieve global recognition, we strive to be a center of excellence in education, research, contributing actively to addressing global challenges, particularly in environmental sustainability. Through international collaboration, interdisciplinary research, and continuous innovation, we hope that Universitas Syiah Kuala will have a lasting impact not only in Indonesia, but also globally. As the oldest university in Aceh and one of the leading institutions in Indonesia, Universitas Syiah Kuala is recognized for its strength in various research fields, including biology and environmental sciences. With state-of-the-art research facilities and highly qualified faculty members, we aim to become a leading hub of scientific inquiry in the region. Our research program have made significant contribution from local environmental solutions to developing policies that support sustainability. Distinguished guests, ladies and gentlemen, Universitas Syiah Kuala is dedicated to the first ICBS conference theme, Aligning Biodiversity and Climate Initiatives for a Sustainable Future, is timely and urgent. Climate change profoundly impacts, making it essential to work together on integrated approach that consider ecology, social, and economic dimension. Biodiversity plays a crucial role in both adapting and mitigating climate change, and maintaining a balanced ecosystem is crucial in preventing for the environmental degradation. Ladies and gentlemen, I hope that this conference serves as a platform for us, for scientists, practitioners, policymakers, and students to discuss, share ideas, and create tangible solutions for global environmental challenges. As we begin, I congratulate everyone involved in making this conference possible. Might inspire you, strengthen your network, and produce actionable recommendations for global environmental policy and practice. Thank you so much. I wish a fruitful conference. Thank you again. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you to Professor Dr. Insinyur Marwan.
To close our opening ceremony, let us now recite a prayer.
We would like to invite Irfan S. to the stage.
Assalamualaikum warahmatullahi wabarakatuh.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم الحمد لله رب العالمين حمدا شاكرين حمدا يوافي نعمه ويكافئ مزيده يا ربنا لك الحمد كما ينبغي لجلال وجهك الكريم وعظيم سلطانك اللهم صل على سيدنا محمد وعلى آل سيدنا محمد والحمد لله رب العالمين ربنا اغفر لنا ذنوبنا ولوالدينا وارحمهما كما ربيانا صغارا ولجميع المسلمين والمسلمات والمؤمنين والمؤمنات الأحياء منهم والأموات برحمتك يا أرحم الراحمين اللهم إنا نسألك سلامة في الدين والدنيا والآخرة وزيادة في العلم وبركة في الرزق وتوبة قبل الموت ورحمة عند الموت اللهم هون علينا في سكرات الموت والنجاة من النار والعفو عند الحساب ربنا هب لنا من أزواجنا وذرياتنا قرة أعين واجعلنا للمتقين إماما ربنا لا تزغ قلوبنا بعد إذ هديتنا وهب لنا من لدنك رحمة إنك أنت الوهاب ربنا ظلمنا أنفسنا وإن لم تغفر لنا وترحمنا لنكونن من الخاسرين وسلام على المرسلين والحمد لله رب العالمين الحمد لله رب العالمين السلام عليكم ورحمة الله وبركاته.
Thank you to Irfan S.
Next, documentation sessions. We kindly request the presence of Professor Dr. Insinyur Marwan, Professor Dr. Budi Setiadi Daryono, Profesor Dr. Taufik Fuadi Abidin, Dr. Insinyur Wiradharma, Mrs. Esi Harneli PhD, dan Profesor Dr. Leni Fitri to come to the stage. Okay, the first photo will be taken in a formal pose. We count 1, 2, 3. Okay, the next photo will be taken in the USK Unggul pose. We kindly ask everyone to make a U sign. 1, 2, 3.
Okay, next, we kindly request the presence of Professor Dr. Suhartono and Mr. Rudi Hadiansyah Putra to come to the stage.
The next photo will be taken in the USK Unggul pose again. Oh, okay, thank you.
Next, we kindly ask all honored guests to stay in the front to return to their respective position while remaining standing, as we will take a group photo with all participants. We also request all participants to stand.
Okay, the first photo will be taken in formal pose. We count 1, 2, 3. Okay. The next photo will be taken in the USK Unggul pose. We kindly ask everyone to make a U sign again. We count at 1, 2, 3.
Okay, thank you so much to the honored guests and participants. Please have a seat. Oh, we're so sorry. Let's do a Salam Lestari pose. In 1, 2.
Dear guests, the opening ceremony has officially ended, and we now move on to the coffee break sessions. Feel free to enjoy the snacks and drinks that have been prepared on the right and the left side of the room.
We kindly remind all participants to keep the room clean by throwing the trash in the bins provided.
The coffee break will be last for 15 minutes, so please return to the room afterwards as we will continue with presentations by our keynote speakers.
Kuala. We extend our warmest greeting to all participants from distinguished universities across the globe who have come together to play a crucial role in advancing the field of biological sciences at the first International Conference on Biology and Environmental Science. Your collaboration, knowledge exchange, and innovative ideas are key to driving environmental sustainability and the progress of biological sciences in the country. This international conference and our community engagement efforts strengthens our bonds and generate positive impacts that resonate nationally. We wish you the great success with the conference and the COBE 2024 work meeting. May this series of events serve as an inspiration and bring lasting benefit for us all. We hope you enjoy participating and may everything proceed smoothly and successfully. Wassalamualaikum warahmatullahi wabarakatuh.
Assalamualaikum warahmatullahi wabarakatuh. Welcome to Biology Department, Faculty of Mathematics and Natural Sciences, Universitas Syiah Kuala. We are proud to welcome participants from Indonesia's leading universities who have come together to collaborate in advancing the biological sciences. The International Conference on Biology and Environmental Science serves as a valuable platform for academics and scientists to jointly develop strategies, share knowledge, and foster innovation aimed at environmental sustainability and the advancement of biological sciences in our country. This international conference, along with our community service initiative, enhances our synergy and brings tangible, positive impact to our nation. Congratulations on the successful implementation of the COBE 2024 work meeting. We hope this series of events inspires and brings significant benefits to all. Enjoy your participation and Assalamualaikum warahmatullahi wabarakatuh.
Assalamualaikum warahmatullahi wabarakatuh. Welcome to Biology Department, Faculty of Mathematics and Natural Science, Universitas Syiah Kuala. We excited to host participants from Indonesia's leading university for the COBE Works meeting and the first international conference on biology and environmental science. Congratulations on the successful COBE 2024 work meeting. We hope this event inspire and benefit everyone involved. Enjoy the conference, a platform for collaboration in advancing biological science and promoting environmental sustainability. We gather here because we care about tomorrow. Please enjoy. Assalamualaikum warahmatullahi wabarakatuh.
For the invited guests and ICBES participants first. And we also kindly want to invite all the committee of ICBES 2024 to come to the stage to do a documentation sessions. We repeat, we want to invite all the ICBES 2024 committee to come to the stage to take documentation session.
We remind you guys to all the community, please to come to the stage because we want to take documentation sessions.
is one of the departments in Faculty of Mathematics and Natural Science, Universitas Syiah, Kuala. Inaugurated on 17 February 1989, the Biology Department continues to develop its existence. This is proven by Unggul accreditation title that has been obtained by the Biology Department since 2023. Until now, the Biology Department has supervised 2 study programs, bachelor's and master's degrees. The Biology Department continues to be committed to developing science through education, research, and community service in order to create graduates who can compete globally.
The Department of Biology has a vision to become a center for the study of biology oriented towards tropical biodiversity in Indonesia, and to provide graduates with innovative and independent socio-entrepreneurs at the global level.
In order to carry out its vision and mission, the Biology Department is supported by teaching staff that graduate from various universities in the world, with 3 professors, 7 associate professors, 18 lecturers, and 4 associate assistants. The Biology Department is qualified to develop science. The Biology Department now has several laboratories, including basic biology, ecology, microbiology, physiology, structure and development, genetics and biomolecular, and biosystematic laboratories. Lecturers and biology students are actively involved in various activities such as community service, seminars, training, and field activities that support the learning process.
The research at the Department of Biology aims to solve a number of actual problems, from ecosystem restoration to innovation in biotechnology.
We collaborate with various institutions to create synergy between science and society. Our alumni work in a variety of sectors, from research, education, and environmental conservation.
Hi everyone, I am Aigia and I am a biology alumni. Being a biology student taught me to think critically and recognize the complex connections in nature, providing my foundation essential to my growth from fieldwork to lab experiments. The experiences I had shaped me in ways I never expected. The skills we developed have empowered us to make changes in research, in education, in industry, and beyond. Biology isn't just a subject, it's a way of contributing to the world.
Join us at Department of Biology, Faculty of Mathematics and Natural Sciences, Universitas Syiah, Kuala, and be part of the solutions for the future.
Welcome back to ICBS 2024. Dear guests and participants of ICBS 2024, we kindly ask you to return to your respective positions as we will continue the event.
We will now move on to the presentation sessions by our keynote speakers, which will be moderated by Dr. Insinyur Wiradharma.
Dr. Insinyur Wiradharma is the Deputy Dean III of the Faculty of Mathematics and Natural Sciences, Universitas Syiah Kuala. His research interests particularly in the field of biology conservation.
We now invite the moderator to take the stage.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning everyone. Welcome to the first ICBS conference. My name Wiradharma and I am honored to be your moderator today. We have an exciting session ahead with two distinguished speaker who will share valuable insight into key topic in biology and environmental science. Give you applause for our speaker. I start for introduction for the first speaker. Our first speaker join us with Zoom is Professor Dr. Insinyur Iskandar Z. Siregar, IPU ASEAN M. And let me brief CV from Professor Dr. Insinyur Iskandar Z. Siregar. Professor Dr. Iskandar Z. Siregar is expert in forest genetic, tropical silviculture, tree improvement and sustainability. He earned his bachelor degree from IPB University, his master from University of Canterbury, and his doctor degree from George August University Zoo, Göttingen. His research focus on ecological restoration, global timber tracking and ecological society economy, and of course tropical lowland rainforest transformation system. He is also involved in CRC 990 project, which look at sustainability of tropical forest. Today, he will present on democratizing sequencing technology for biodiversity research. Okay, please join me in welcoming for Professor Dr. Iskandar Siregar and Mr. Professor, time is yours. Is it clear, my sound, my voice, Mr. Professor? Yeah, Zoom. Mic is not open yet. Pais, your mic is not open yet. Your microphone is not open yet, Pak Iskandar. Your microphone still mute, Pak. All right, it's work.
Let me first share my first slide here. This is about my presentation. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Thank you, Pak Wira, for moderating my session this morning. So, first of all, I would like to congratulate the organizing committee for conducting this very important seminar, ICS 2024. And also the Honorable Director, Syiah Kuala, in Fakultas MIPA and the Chairman OB, as well as the organizing committee ICS 2024. My talk today is about the democratizing sequencing technology for biodiversity in these diseases. Our work was our experiences for the last 4 years after the COVID. Yeah. So we tried some new technology that allow us very expensive and difficult to access, and we have to submit our sample overseas. Our work based on the research support. This is a title of my presentation, the introduction, including democratization of technology, as well as practical application, and also the closing or recent lesson learned. Ladies and gentlemen, as an introduction, the wealth of tropical biodiversity, especially in Indonesia, has attracted many scientists all over the world to carry out both basic and applied research, including the potential bioprospecting. In this case, is when we are talking about bioprospecting, there will be a commercial use of from the research. From the biodiversity research, we know that we are Indonesia subscribed to different global agreements such as Convention on Biological Diversity, Cartagena Protocol, Nagoya Protocol, etc. And we learn also from the previous experiences that some cases happen in the in the scientific world, where misuses of biological materials happen because good research practice were not followed due to mostly lack of understanding and compliance from our partners, for example. And the challenge is now how we can do good implementation of research, international research, as well as the second one, finding good ways to utilize the affordable technology. Affordable meaning is we can pay it and we can access it easily. From the data of Scopus database, I checked different terms on biodiversity research, and it linked to the SDGs 15. One of them, life on land. Biodiversity also, of course, linked to other SDGs. For example, SDG 14, life below water. But from life on land, we see that still biodiversity research is important, but it is maybe due to different drivers like land use change, land conversion, landscape research, etc. And when we are talking about the land, especially Indonesia, we know about the different kind of protected areas. In this case, is very important. I think where are the national parks? I think in Aceh we know Gunung Liser National Park, one of the example, very large one. And currently, based on the latest data, there are 54 national parks in Indonesia, crossing western part Indonesia as well as eastern part Indonesia. And we know that from the previous publication, according to the SEAL 2018, that in this region, especially Wallacea region, as well as Southeast Asia region, there are still common plant species which has been identified, as well as can be found where they have large distribution, as well as very small distribution. And of course, technology are really needed, yeah, apart from the regulation, like different law on biodiversity conservation. We know also the importance of the role of technology for dissecting different kind of nature properties or nature characteristic. And we know the development of technology is there since 1950. For example, we know about the sonar technology, including the current technology, especially on the sequencing technology, which has been initiated during the 2010 to 2015, and now we are entering to the future technology or the future practice. And we have to be adaptive as a researchers in order to work more effectively, as well as work more efficiently. And there are trends, of course, because biodiversity research are very important. A lot of people are discussing is, apart from carbon, carbon may be already a little bit mature on how to do valuation, for example, with different kind of carbon financing system. But for biodiversity, we are still discussing, especially about the biodiversity credit, how we can make this biodiversity become useful industry, which can be accessed by many parties. Yeah, start from the farmers, for example, until a multinational company. And McKinsey has written a report on the importance and the role of biorevolution in the future, yeah, because of the technology development. And a lot of industry will emerge, I think, including industry on biomolecules, biosystem, biomachine interfaces, as well as biocomputing. And we know this week, I think, and last week, at the moment, I think already concluded there is COP16 at Cali, Colombia. And I noted from the outcome of the meeting, and there are, of course, several negotiation. Yeah, one of the important regarding to my talk this morning is about the Cali Fund, which is launched about the sharing the benefit from digital genetic information. And I think it has been agreed, yeah, with other negotiation topics. Yeah, in the relating to digital sequence information, I think DSI, yeah, especially when it is used by the large company or other major entity, they have to contribute to the Cali Fund based on a percentage of their profit or revenues. So the model has been agreed, target larger companies that most reliant on the SI. And developing world countries will benefit from a large part of this fund, with allocation to support implementation, Kunming and Montreal biodiversity framework according to the priorities of government. So we have to work on it, waiting, and the mechanism really important to support the future biodiversity research, especially in Indonesia. And to be prepared, I think we need to adapt also to be acquainted with different kind of technology from the history. I think for the professor or senior lecturer who work for the past fourth year, they experiencing different kind of sequencing technology from the first generation sequencing technology using the normal Sanger sequencing, the second generation using Illumina, PacBio, as well as the third generation sequencing with generate long read, especially from the current anion sequencing produced by Oxford Nanopore. This is the characteristic of its sequencing technology from long read sequencing, including accuracy, including high throughput as well as accessibility. And our group in Bogor select the easily accessible technology like Oxford Nanopore technology, starting with the Minion model. So we successfully first sequencing our sequencing work in 2020, when the COVID just started, and we learn from this research how to generate the long read data as well as how to analyze the data using different bioinformatic platform. Ladies and gentlemen, this is the dry and wet lab workflow for Oxford Nanopore platform that we experiences. From the first one, library preparation. So we have to prepare in this work different kind of small equipment. When we ready with this library preparation, so we found that the use of qubit to quantify the DNA concentration is very important. Qubit instead of nano drop, for example. So we learn from this research. And then we do sequencing, of course, after we prepare library. And finally, we do the bioinformatic analysis from base calling until annotation. So that kind of workflow has been developed in our lab, and we apply it to many different tree species, and now it's expanding not only to the species but also to the insect. This is the work that we are doing. Using this technology, we can do sequencing anywhere, basically, in the field, including in the field, in the office, in the hotel, etc. This is the for example, after coming back from the field, we do a sequencing in the office, using the different samples. So in this research, we are doing the sample from termite, for example. So this is the result from our exercise using this technology. Otherwise, we cannot do it using the very expensive sequencing machine. So we just do a sequencing of different tree species from Diospyros, ebony, balsa, agarwood, pometia, rambutan irian, kapur, kayu besi, or ulin, Castanopsis, and more tree species has been sequenced and to be sequenced in the near future. So this is a set of the equipment that we are needed if we are going to the field, I think, from the portable bento DNA machine, centrifuge together with the PCR machine. All are portable, that can be brought to the field, and we can do research with our student directly, and we do sequencing later in our room, for example. This is the result of our work, using portable sequencing to support, for example, understory vegetation study, especially for species identification, our student can learn how we can identify correctly species using the data generated by the portable sequencing in this case, apart from normal herbarium or morphology identification. Yeah, students start to do their bachelor thesis for the first time, and they learn about from the DNA extraction, doing DNA library preparation as well as sequencing, including data analysis until DNA annotation and do phylogenetic studies, for their research. So another step is from the partial genome assembly, we move on to the little bit complete chloroplast genome. For example, this is from Cinnamon burmanii. We successfully resequence the complete chloroplast from Indonesia, for example, to add the data in the global database. As we know, there are now 3 global database: European Nucleotide Archive in Europe, DDBG in Japan, and NCBI in US. We send currently our data, raw data to DDBJ in Japan. This is for Santalum album. Basically, we can do different sequencing and create our own chloroplast genome, and our student can understand the gene one by one to explain different mechanism, including future prediction on metabolites to be resulted from this DNA. We also now ongoing to do a timber tracking. We finished with the sequencing of kayu ebony in Sulawesi using this technology, and we still ongoing the work, not only genetic but also wood anatomy, isotope analysis, as well as chemical analysis. So DNA technology can be complementary to other technology. Very important to the capacity building, not only for our student, but also for high school student around Bogor. Yeah, we conducted different webinar for guru biology or biology teachers, high school around Bogor, to experience the real work on extracting DNA, analyzing data, and interpreting the data. The next step that we are doing is we collaborate with the ORG one, which facilitate the sequencing work on critically endangered species, according to IUCN. So at the moment, we completed the sequencing of critically endangered of Dipterocarpus littoralis, which are endemic to Pulau Nusa Kambangan. And also with the result like this one, so we generate quite a big data. The problem is now how to analyze the data. Yeah, this is the real bioinformatics work. The second one that we manage also to sequence the Mangifera casturi. Yeah. This is already extinct in the wild actually, but still be found in the garden of community in Kalimantan. Ladies and gentlemen, from our work we learn different lesson. Yeah, very important is to prepare the capacity of resources. Which work interdisciplinary. So we need the real biologists, we need bioinformatician, and also ethnobotanists to work together. And we need to create a pilot project and working together And we need, of course, to prove that our work has been acknowledged to publish our paper, very important. It brings spirit if we publish our paper from our existing work. Of course, we need to disseminate the protocol and expanding the network. Again, improving continuously the capacity because the technology developed very fast, including the bioinformatic platform. There are always new things happening. At least to take ways to be implemented in the future, we need to shape up our research roadmap, including introducing the technology to wider potential user for more democratization. So democratization is there. Now is everything is dependent on us. I think that's all my presentation. I hope it will be, let's say, inform you about what we are going to do in the future and how we can network. We hope that Kobi can facilitate in the near future. Thank you so much. Wassalamualaikum warahmatullahi wabarakatuh.
Regard for your excellent presentation. It was both enlightening and inspiring. So because we have two speakers for today, and then this is the panel system. Yeah. For the next speaker, by the way, I am going to tell you about Prof. Iskandar, also as a Vice Rector for Global Connectivity Cooperation and Alumni Affairs. Okay, everyone. Next for the second speaker. Please welcome Professor Dr. Suhartono, MSc, to the stage. Give you applause for him. Our next speaker is Professor Dr. Suhartono, MSc, from Department of Biology, Faculty of Mathematics and Natural Sciences, University of Syiah, Kuala, Banda Aceh. Let me brief CV of Professor Suhartono. Professor Suhartono, MSc, has been a lecturer and researcher at Syiah Kuala University since 2006. He earned his bachelor degree in biology from Bogor Agriculture University, completed his master in medical and molecular microbiology in University of Manchester, UK, and his doctorate in cell and molecular biology from University of Arkansas, USA. He teaches microbiology related course, and his research focus on spread of antibiotic resistant gene in aquatic system through horizontal gene transfer. Today, he will present on topic antibiotic resistance in aquatic system from urban runoff resistant microbes. Please welcome once again for Professor Suhartono, and Professor, time is yours. Thank you very much.
Baik, thank you Pak Vieira for the introduction. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. So good morning everyone. Today I'd like to give a talk related to antibiotic resistance. So I'm going to talk about how the antibiotic resistance is happening. It starts from ourselves, from our activities, so that lead to make the bacteria in environment, specifically in the aquatic environment, to be resistant. So as introduced before, my name is Suhartono. I'm a lecturer and researcher in the biology department. MIPA, Universitas Syiah Kuala. I was working in the department since 2016. So it's approximately, it has been 17 years now. So this is a short CV. So basically, my research focused on antibiotic resistance, about how the genes that are responsible for the resistance to be disseminated, and how the gene makes some bacteria to be persistent in the environment. And also, this is about the outline about my talk. So basically, I'm going to discuss again related to the what is antibiotic resistance, how it happens, and also I'm going to talk about how the resistance, how the pathway that make the bacteria to be resistant, specifically bacteria in the environment, in the aquatic system. And I'm going to talk about a little bit about horizontal gene transfers, a phenomenon that make bacteria to be resistant easily from one bacteria to another bacteria. And I'm going to relate it about the selective pressures, or also some sub-inhibitory level that can make bacteria to be resistant. And the rest of the talk about the public health impact, and also about the strategy, how to cope or to prevent the antibiotic to be more threatened. Okay, next, this is the introductions. I believe all of us here, we have been consuming antibiotics in many ways, from the oral digestion, from the injection, and so on and so forth. So basically, the antibiotic resistance is defined as the ability of the microorganisms or bacteria to withstand the effects of antibiotics. Basically, there is a pathogenic bacteria when it gets antibiotic in their surrounding, they're still alive. So basically, they can be resistant to the antibiotics. So if we back to the history where the antibiotics were firstly found back to 1940s, just few after that, the bacteria become resistant to the newly invented antibiotic at that times. So like for example, in 1940, we found penicillin, and like 3 or 4 years after that, they found some bacteria to be resistant to the penicillin. So basically, antibiotic resistance is natural phenomena that it happens naturally. Like it or not, it happens. The problem is the human activities make it happen quicker than before. So there is human activities make the antibiotic resistance become quicker than it's normally happening. So some scientists projected that in the future, if we don't do nothing, there will be antibiotic resistance will be the #1 top cause of the death by 2050. So it's more than even cancer in 2050 as if we do not have any interventions at this current moment. So this is the projection. So AMR, it stands for antimicrobial resistance. On the list, it's #1, number one. That will be the top cause of the leading cause of death of people projected in 2015 by 10 million per year. So people will be dead by the bacteria that infect them, that are resistant to the current antibiotics. If we don't do nothing about it, they will die. Okay, so there will be like they're dead because of the bacteria is now resistant to all antibiotics that we have at the moment. So this is the simple mechanisms, how the antibiotic works and how the bacteria cope with the antibiotics. on the left-hand side, so we know that some antibiotics that can destroy these bacteria cells with some various mechanisms. Some antibiotics can kill the bacteria through the cell wall, the inhibitions of the cell wall synthesis, for example, like beta-lactam antibiotics that we know, penicillin, cephalosporin. The drugs can kill the bacteria by inhibiting the cell wall synthesis. The other mechanisms, some antibiotics can inhibit the protein synthesis, and another antibiotics, they can inhibit the nucleic acid synthesis, inhibited the synthesis of the metabolites, and also inhibited how the cell membrane of the bacteria is formed. That's how some antibiotics can kill the bacteria that infect people. On the other hand, the bacteria, the pathogenic bacteria, fights back. How they do have a fight back? They develop some resistance. So they have some mechanisms how they fighting back. For example, some bacteria can develop the efflux pump. So basically, if the antibiotics come into the cells, the bacteria will export the antibiotic outside the cell by pumping them outside the cell. That's the first mechanism called efflux pump. The other mechanisms is by inactivating the antibiotic substances. For example, antibiotics, beta-lactam antibiotics like penicillins, when they come into the bacterial cells, the bacteria will synthesize another enzymes called beta-lactamase that can inactivate the beta-lactam of the antibiotics. So the antibiotics can no longer active within the bacterial cells. Other mechanisms, they can have bypass the target or make some modifications of the antibiotic targets. So basically we have antibiotics on one hand, and the bacteria have the other mechanism to fighting back of those antibiotics. So this is what Paul Ehrlich says in early 19. So basically the Paul Ehrlich says that drug resistance follow the drugs like a faithful shadow. What's the meaning of this? The meaning is, like as I said before, like the antibiotic resistance is naturally occurring. Like it or not, just like it or not, it happens. When we find a drug, there will be some antibiotic resistance towards the drug that we justly found. So basically, it's just cat and dog. It's like a race. It's just like a rat race between the antibiotics, human find the new antibiotics, and the bacteria finding a new ways to fighting back of those drugs that we have found. So this is the typical how the bacteria become resistant anyway. So there are various mechanisms how the bacteria become resistant. The first one is on the top left-hand side. It's called natural antibiotic resistance. Some bacteria, they are naturally resistant to the antibiotics. We call it like intrinsic antibiotic resistance. For example, let's say some antibiotics kill the bacteria by inactivating the key enzyme. But the pathogenic of the bacteria do not have those enzymes. So basically, the antibiotic won't affect to those bacteria because basically they don't have the target of the antibiotics. So it's called the natural or intrinsic antibiotic resistance. On the B part, it's called the acquired antibiotic resistance. Basically, one bacteria can be resistant by receiving the antibiotic resistant gene from the other bacteria. This is happen when we have the horizontal gene transfer. Basically, one bacteria resistant, they share the antibiotic resistant gene to other bacteria that now susceptible and then now become resistant. So basically, it's like sharing the genes. The third one is like the C part is called situational antibiotic resistant. So basically, antibiotic resistant can be situational. We know like some bacteria, they can live planktonic, which is cell, and then they can have make a group called biofilm. So basically, when they are in planktonic form, they are susceptible to antibiotics. But when they are forming the biofilm, they become resistant. This is what we call as situational antibiotic resistant. And also, do not forget that we have some separations talking about the antibiotic resistance. Some bacteria are susceptible, the other bacteria are tolerant, and the other bacteria are resistant. So basically, it's just a spectrum between susceptible and also the resistance. We have tolerant. What does the tolerant bacteria means? So basically, the tolerant is bacteria that is susceptible, but it takes some time for the antibiotics to kill them. So for example, for the susceptible bacteria, when there is an antibiotic can instantly kill them. But it's not happening for the tolerant. It's susceptible, but it takes more time for the antibiotics to kill them. That's for the tolerant. And the third one, of course, the resistance. The existence of the antibiotics do not have any effect on them. So basically, this is how the antibiotic resistance is happening. Okay, next. So I'm talking about the previous one, I'm talking about the horizontal gene transfers. Basically, in bacteria, they can share the gene by transmitting the gene from one bacteria to another. Not only from the same species, but also they can transfer these genes from different species. There are three mechanisms that we have known about the horizontal gene transfers. We know like some bacteria can have transformations, the other bacteria some transductions, and the other one have conjugations. The three processes that can describe the horizontal gene transfer. Next, but let's not forget, some bacteria also a feature called mobile genetic elements that can make the horizontal gene transfers it's easily happening. What is mobile genetic elements? So basically some bacteria, they store or they code the gene within the other DNAs. They can be encoded in the plasmid, they can be encoded within integron, also transposons. So basically this called the mobile genetics elements that can easily transfer from one bacteria to another bacteria through horizontal gene transfer. Next. So what's the deal with the aquatic environment? Why the topic is about the aquatic environment? Why don't we talk about the clinical settings in the hospital? So this is important. The aquatic environment is the reservoir for all of our activities. For example, if we consume the antibiotics, what happens to the antibiotics? Some of them will be absorbed to the body through our blood. What about the rest? They will be excreted, right? Through what? Through urine, right? Urine and also the feces, the fecal. What happened to the urine and the feces? They goes to the septic tank or sewers, and by the end of the time, they will end up in the aquatic environment, in the river, or back to the sea. It happens from one person, and also it happens for the clinical settings. So basically Okay, we can go next. So where does the antibiotics comes from? It's from the pharmaceutical company, right? They make the antibiotics. Okay? So once the antibiotics are produced, what happened to the antibiotics? We use them. Who use them? For human, right? For human treatment. The other thing, we use antibiotics to treat the animals, right? This is another fact. The more the antibiotics are mostly used to treat the animal than the human. So one third of our antibiotics is for human treatment. The 2/3 is for animals. The animals that use for cattle, for chicken, for animals basically. So after they use, how they will end it up. So for human, for human treatment, they goes to the urine or fecal, they goes to the wastewater treatment plant. And for the animals, they go to livestock treatment, and also some of them are for aquaculture. And then they go to the soil, and some of them become sludge. And after that, they go to the groundwaters and also the surface water. So basically, in aquatic environments, they'll contain the antibiotics both from human and also from the animals. So it accumulates the antibiotics in the aquatic environment. So how the aquatic environment receive their antibiotics comes from. So the number one, the top list, is from industrially polluted surface water, is the top one. And the second one is from hospital effluent. The third one from the municipal sewage or wastewater treatment plant. And also from municipal sewage and from the rivers and also from the sea. This picture shows the antibiotic concentrations within the source. For example, the sea, the water in the sea, they may contain antibiotics in it, but the concentrations is very low. It's 0.1 microgram per liter, meaning that if we took seawater, we took 1 liter of the seawater, they may contain 0.01 microgram inside it. So this is the typical MIC. What is MIC stands for? It stands for minimum inhibitory concentrations. The concentrations that can inhibit or kill the bacteria. So basically what happens with this picture, the antibiotics in our environment, in aquatic environment, most of them are below the MIC level. Basically, if we have some bacteria in the aquatic environment, they are exposed to the antibiotics with the concentrations that do not enough to kill them. This is, we call it L sub-inhibitory level. Or, to be exact, to be daily exact, we call it underdose. Okay? So basically we have pathogenic bacteria in aquatic environment. They are exposed with the antibiotic from these sources, but the concentrations is not enough to inhibit or to kill them. So what happened to the bacteria? They start to make some adaptations. They make some modification so that eventually they become resistant to the antibiotics. This is what happened. Next. So this is basically talking about the sources and the selective pressures and also how their resistance occur. Okay, next. This is another quote from Alexander Fleming, who found the penicillin. What he said, it says that it is not difficult to make microbes resistant to penicillin in the laboratory by exposing them to the concentrations not sufficient to kill them. Right? This is what happened. For short, we call it underdose. If we consume antibiotics, when we visit the doctor, we got some infections, and we visit the doctor will make some prescriptions about the antibiotics, and when we start to consume them, and after a few days, we feel better, and we stop consuming it. At that moment, we make the bacteria inside the body become underdose of the antibiotics. This is what happened. What Alexander Fleming said about this. Basically, it talks about the underdose. And the underdose also happening in the aquatic environment. Why? Because in the aquatic environment, they contain antibiotics from many sources. From the wastewater treatment plant, from the houses, and so on and so forth. But the concentration is not enough to kill them. So the bacteria try to make some adaptations by modifications, by mutations, and they become resistant. So what happens? So after this, what are our strategies? Of course, we need some interventions. If we do not do nothing, what it has been projected by 2050, there will be some 10 million deaths because antibiotic resistance is going to happen. So the intervention is this, like we can go from science and technology. If bacteria become resistant to the current antibiotics, so basically we find a new one, right? But there is a trade-off of that, because if trying to find a new antibiotics is not an easy. It takes time. Or we can go by educations. We can go by educations, by giving people awareness. So when we try consume antibiotics, we make the right dose. We do not want to make underdose. And we consume the right antibiotics, right? Make sure that we consume the right antibiotics. Because if we try to consume the wrong antibiotics, they also become resistant. Okay, next. Yeah, this is some our works related to the antibiotic resistance in aquatic environments. We collected some samples, water samples from the rivers, and we found that some gram-negative bacteria, E. coli or coliform, to be resistant to the antibiotics. Next. Yeah, this is another our works that have been published previously. Next. Yeah, this is also another our works about the isolations, some bacteria in the water aquatic environment. Next. So this is the take-home message for our talk. So our antibiotic resistance in water system is a complex, is an interconnected challenge requiring immediate attentions. And also it comes from multiple sources, from the hospital, from farms, and from urban area. And also they need some strategies to deal with those for those antibiotic resistance. And the last one, next. So this is another code for antibiotic resistance. This is the concept. So what doesn't kill you makes you stronger. It happens with antibiotics, antibiotic resistant bacteria. if they are exposing with the antibiotics, which is underdose, which is low below the concentration that are enough to kill or to inhibit them, they become resistant. So this is what doesn't kill you makes you stronger. It happens to our lives also, right? We become stronger with all the challenges that we have. It's also happened with the antibiotic-resistant bacteria. Thank you very much. I'll give back to Pak Wira.
Thank you very much, Professor Suhartono. Thank you for insightful presentation. Now we will begin Q&A session. If you guys have any question, please raise your hand, and then we will first address to Professor Iskandar, and the second one for Prof. Suhartono. Okay, we give for one first session to 3%, I think. Raise your hand and introduce yourself, name and department or university or something else. Does anyone? Yeah, okay. One. Second one. And give address for which speaker, right? Anybody else? Okay, two. Stop two, right? Okay, give it simple and then address for which speaker, okay? All right, please.
Okay, Assalamualaikum warahmatullahi wabarakatuh. Thanks for the attention that has given to me. I'm Della Puspita and I'm a student from biology. I would like to ask a question that's bacteria who there in white blood. Why I ask these questions? Because I heard something new that's from my siblings. In the last week, I have a siblings. He is 3 years old, and after that, the laboratory results said our siblings there is white blood in his. There is a bacteria in his white bloods, and after that, he got a fever until make his be convulsions. So I would like to ask you that who causes the bacteria there is in white blood? Thanks for me. Wassalamualaikum warahmatullahi wabarakatuh.
Can you explain or introduce yourself? Where are you from and okay, udah tadi ya? Siapa namanya tadi? Okay, the second one. Lupa tadi.
Bismillahirrahmanirrahim. Let me introduce myself. My name is Salimina Ulfa, a student of Biological Science, University of Syiah, Kuala. I have a question for Professor Suhartono that in what condition we can to consume antibiotic and what we have to do if we already consume it to prevent the resistance of bacteria in the environment. Thank you.
Okay, enough. So we have two questions and then address all the question to Mr. Professor. All right, time is your Mr. Professor.
Yeah, thank you, Pak Wira. So I've got two questions actually. The first one is how the pathogenic bacteria can be found in our blood cells. Is that correct questions? So basically, and when pathogenic bacteria infect the body, right, they can go to the blood through our transportation systems. That's why we have a term called bacteremia, which is the existence of the bacteria within our blood systems. So the white blood, if we take some blood samples, we can find the white blood obviously we find the red bloods, and also we found the white blood cells, which is the guardians of our immune system. So basically, that's the general term. So basically, if the bacteria, when they start infecting our body, they can go through our transportation systems through the blood. It's called the bacteremia, the existence of the bacteria within our blood system. The bacteria, and by the way, the bacteria is originally sterile. So there is none of the bacteria should be in the blood. That's why the blood can be a diagnostic, one of the clinical sample that can we take to detect the existence of the bacteria. If we found a bacteria in our bloods, the meaning of it, the patient is suffering from the infections because originally our blood is sterile. No bacteria can live in our blood system. So if they found the bacteria in the white blood, meaning that it's normal when we got some infections. So we took some blood, we get the red blood, or also the white blood. So we took all the bacteria inside. And the second question is about the antibiotics prescriptions. Yeah, when the doctors make some prescriptions about the antibiotics, we need to make some confirmations. If you go to the hospital suffering from infections, what does the hospital do? First time, they will give you some general antibiotics. And that time also, they take some clinical samples to make sure what is the bacterial pathogen that infect the patients. Remember that if we want to identify the bacteria, it takes time, right? They need to take the clinical samples, bring to the lab, and then grow them on the media, and we need some time to incubate them until the hospital can exactly know what is the bacteria. Once they know the bacteria, they will give another antibiotics that are suitable for the bacteria. Because the hospital has the data, what is the bacteria that are susceptible from. For example, if Pseudomonas aeruginosa is detected in your clinical samples, so basically the hospital will they have a list what are antibiotics that can be used to treat the Pseudomonas that are still susceptible of the antibiotics. So they'll change the antibiotics from the general one, the broad spectrum, to the specific ones. Okay. Another thing is like make sure when you consume antibiotics, it's the right dose. So finish all the antibiotics that are prescribed for. Finish all of them, regardless your body conditions. Sometimes you feel better, right? You feel better. You don't need any more antibiotics, right? But let's not forget that there is a condition called asymptomatic. Asymptomatic is that you are currently infected, but there is no sign. You don't feel any fever and so on and so forth. But basically, technically, you are still infected with the bacteria. So we want to take all the antibiotics at the right dose, so make sure they can inhibit or kill them. Maybe that's the answer.
Thank you, Professor. So we have applause. Give applause for our professor. Does anyone have a question for the second session? And point me which speaker that you are addressed for the question. Anybody else? No? All right. Okay, if there is no question anymore, okay, big applause for our speaker, Thank you, both of you, Professor Dr. Iskandar Z. Siregar, IPU ASEAN Eng, and Professor Dr. Suhartono, MSc, for their informative and insightful presentation. Thank you for all participants for being part of important session. We hope we found beneficial and inspiring, and of course, see you for the next session. Assalamualaikum warahmatullahi wabarakatuh. Terima kasih, Prof. Kita foto dulu. We have take a picture, right? Where is photographer? All right. Thank you, Prof, for the time.
We kindly ask the keynote speakers and moderator to remain on the stage for presentations of mementos. For Professor Dr. Iskandar Z. Siregar, the memento will be a certificate, and here is the certificate.
For Professor Dr. Suhartono, the memento will be presented by the Chairman of Konsorsium Biologi Indonesia, Profesor Dr. Budi Setiadi Daryono. Okay, for Professor Dr. Suhartono, the memento will be presented by the chairperson. Now we invite Mrs. S.C. Harnelli, PhD, to the stage.
Okay. Thank you so much to our keynote speakers, Mrs. S.C. Harnelli, PhD, and the moderator. We kindly invite everyone to return to their respective seats.
We will now move on to the presentation sessions by our invited speakers, which will be moderated by Mrs. Elizar, PhD.
Mrs. Elizar, PhD, is a lecturer at Mathematics Education Department, Faculty of Teacher Training and Education at Universitas Syiah, Kuala. Her research interests particularly in higher order thinking skills in mathematics, quantitative research, large-scale assessment, and hierarchical linear modeling.
We now invite the moderator to take the stage.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. My name is Eliza, and I will be your moderator for this session. Welcome again to the first International Conference on Biology and Environmental Science. I hope everyone have a good day and looking forward for the next session. So before we call our next speaker, let me introduce him to you all. So he is a dedicated conservationist with over 2 decades of hands-on experience protecting Indonesia's lizard ecosystems. As a trained conservation biologist, he has led initiatives from rhino protection to ecosystem restoration, working with national and local agencies to protect endangered wildlife. He was a former director of Lizard Conservation Forum and now the director of Saman Sedati Lestari, a company focusing on managing ecosystem restoration concession, sustainable production, and ecotourism. Without further ado, please welcome Mr. Rudy Hadiansyah Putra, MSI. Okay, Pak, Mr. Rudy, the stage is yours.
Thank you everyone. Assalamualaikum warahmatullahi wabarakatuh. My pleasure to be here and my respectful to my lecturer here and my senior and junior. So I was studying biology in this faculty, and today I'm working for the conservation in last 24 years, almost 25 years. And today I want to, this is more about the experience. I'm not researcher, I'm not, because conservation and with the research, this is very close, but we can combine this because my focus, how to protect the lizard ecosystem, and then how we can invite the researcher to know and then doing the research in the lizard ecosystem. So today, I will talk about the fighting extinctions, how the species conservation in the lizard ecosystem impactful for the world conservation. So I'm working for the local NGO. Sometimes we call it local NGO or kampung NGO. We don't care because we focus only in the lizard ecosystem. And this morning I read about this. This is all the article about the UN report, how the dangers of the world that every year we loss of the species because of climate change, because of the deforestation, and anything else. And the impact is not only for the biodiversity, but also for the human life in the world. How it's very difficult to find, including for the food now. As we know, food crisis will be happen in many countries, including Indonesia. We import a lot of the rice from the Thailand, from the Vietnam. One aspect because of the biodiversity loss. Next, please. So this is the happen in the lizard ecosystem and the Aceh or everywhere. That's the elephant kill. Or next, the deer. We poach. Next, and the deforestation by the logging. And next, deforestation happens. And we lost so many forests. And World Research Institute calculate last year we lost 3.7 million hectares of tropical forests. But in the UN report also mentioned this is not too late to make a difference. Even we lost so many forests, so many biodiversity, but this is not too late. We still have chance, we have the opportunities to save our earth and to save our life, because sometimes biodiversity is not really impact for us, but the most important thing, because of water, if we protect the forest, if we protect the biodiversity, this is impactful for us because that is our water resource. So that's why we fight. And to protect the forest and to protect the wildlife or the biodiversity. Next, please. And this is another opportunities, but conservation is not about the difficulties. Conservation is about much easier. So that's why we still stand for the conservation in here. And the report said this is 215 million hectares of the forest, of the land, everywhere in the world, can restore by themselves. Can restore by themselves. Next. So this is about agroforestry, and then please next. And the second thing, that is the ecological restoration, is much success than the active restoration. So that's why conservation is not difficult. Next. So this is Kuala Namu Airport in Medan, in the center of Medan, North Sumatra. This is quite far from the forest. And we can see 100 years ago, the Netherlands Dutch colonial, balik dulu, the Dutch colonial boiled the first palm oil in this area. And in 2013, Indonesian government, through the the companies, state companies, boiled this area, changed from this area from the palm oil to be new airport in Medan. And after few years, we can see in the circle the forest growth by themselves. There is no active restoration in this area, and we can see the ex palm oil there. They can grow even quite far from the forest. That's really make me hope how we can protect the user, how we can protect the environmental. That is easier, not difficult. Next. And this is another example. This is ex palm oil in the Leuser ecosystem. In 2009, this is palm oil, illegal palm oil in our forest. And next, please. We restore using the chainsaw. We're cutting down the palm oil, and then, next please, the forest growth by themselves. We don't need any action to restore this forest because of water, because of the wind, because of the animal, bird, and anything else. They can spread those seeds, and then they can grow. So this is amazing of our forest, our land in Aceh especially, and also in Sumatra. Next. And the third, this is how we can use active restoration. This is also ex the palm oil. And then we plant by the human, by the local communities. Next. And the forest also grows. That is simple. So this is our action in the Leuser ecosystem. Next. So this is Leuser. It's quite far from everywhere in the world. And we call this one of the isolated forests in the world. We are very close to Singapore, but need, I think, 22 hours or 25 hours from US, or 13 hours from the Europe. But that's okay, because far, but also very important for the people. Next. So this is the biodiversity hotspot in the Leuser ecosystem. And very important, we can see this, why in this area is important, because lowland forest. as we know, as a biologist, we know lowland with the higher biodiversity rather than in the highland. Our role, know how we can protect in the lowland forest, and then protect the wildlife there, biodiversity, and also for the local, our people, especially for the water. Please, next. So this is the largest continuous forest. We can see if you fly from Medan or Banda Aceh to Aceh Tenggara or the Tapak Tuan, everywhere in Aceh, we can see the beautiful forest. This is lowland forest. Next, highland forest, also green. We cannot see even only one building in this area. This is totally green. 3.5 million hectares of Aceh, still forest. Even some of them is destroyed, but no problem. Next. And also with the very high biodiversity, we have the tiger, elephants, orangutan, and Sumatran rhino. All of these species almost extinct, and we call us the endangered species, critical endangered. And Aceh and Leuser become the last hope for this conservation of these 4 charismatic species. Next, we also have the many other species like hundreds of birds, mammal, reptiles, amphibians, and thousands of the plant. This is really of the biodiversity hotspot, and also as our laboratory for the research. This is really the best and the biggest, I think, laboratory in the world for the biology. So that's why this is our opportunities. Invite the student, invite the researcher to explore the user, not for the explore the mining or the extractive, but we have the very potential for the biodiversity. And the next, for the economic also, because everyone, every expert know about the pharmacy, about the carbon and anything else. This is the source for the economic in near future. In Cambodian, that's a government very clever. They get a lot of money from their forest. And very close, maybe in next 3 or 4 years, there is another alternative economic from, we call it biodiversity credit. That means where the area with very high biodiversity, like in Leuser, in Aceh, we can sell this not as the product, but for the ecosystem services. That is the big compensation later for the Aceh. So that's why I talk to the governments. If we can protect the Aceh forest, if we can protect the biodiversity in Aceh, this is the real big economic impact for Aceh. So we don't need any outsource or anything from the government support, outside support or the debt or anything else, or the support from the international NGOs or international. If Aceh can use this biodiversity, forest, and our water, our clean air for the economic, this is really the big economic in near future. That's why in last few days, I read about the publication that is Prabowo Minister State, that Indonesia potentially get around the 190 trillion rupiah to support the economic from the carbon. Next. So this is Leuser, and we lost around the 500,000 hectare forest in last 50 years. Next. Next, please. Yeah. We have the monitoring system using the, also this is free. I think some of you researcher and then lecturer with the, we call geographical information system. And we can monitor you even with our phone using the Google Earth. and this is free, and we can monitor what happened in our forest for the every day, and we have the special team to monitor what happened every year, every month in our forest. And based on our calculations, we lost 500,000 hectares in last 50 years. This is very big. That's been similar than 400,000 football field. Next. So this is similar than 20% of the forest in Leuser. But I don't want to cry because of the 20%. Our job now, how we can protect the 80% magical forest of the Leuser. I don't want to cry because what for we cry of the 20%, and we publish so much about the 20% of the forest loss. But we forget we still have the 80% of the forest in the Leuser. Next. Next, please. And this is 25 times of the Singapore. And the people in Singapore, they don't know about the Leuser. They don't know about the Aceh forest. What happened if this forest, we cutting down all of this, and then the Singapore will impact for the ecological crisis because they need fresh air. So that's why this is our campaign to them. Even Leuser is closed, but the Singapore have to care about Leuser. And another country also, they have to care. This is not, if we protect the Leuser, that not meaning only for us, the people in here, but also for the people in the world. Next. So, we have the strategy, and we call the first strategy. This is very important for the species. Today, I more specific talk about the species because in this forum because of the biology. But honestly, I never talk about the wildlife for the local communities because they said, what for? Biodiversity for the local communities, they don't care about this because so many orangutan still in Leuser, so many tiger, and making a serious conflict, and also for the elephants. We always talk for the local communities, for the indigenous people, that is, we need to protect the forest because of water. There is no one can live without the water, I believe, and it's my guarantee. We can maybe survive for only 1 or 2 days without the water, but after that we will die. So this is our campaign to the local communities. We introduce, that is, if without the water, there is no life. So that's why we need to protect the Leuser. And we call this wildlife protection team because, you know, many people will cry because of the orangutan. Many people will cry because of the elephant die. And so that's why this is sexist and easy to publish about the wildlife and donor. This is more about the donor attract, but we not really focus on the wildlife. And we call this Wildlife Protection Team, WPT. And this is our work, and very hard work, how we cross the river. This is 5 rangers walking in the forest for the 15 days. There is no vehicle, no anything else. Just keep walking. But don't worry, they're still alive. They still survive. And because forest is very heavy, we love in forest because fresh air. We can swim in the very nice river. We don't need the very big food, very fancy facilities because what for? We cannot bring it. So only with the simple, but we need around the 25 kilograms for the 15 days. This is very hard for you, I believe. So that's why only the trained people can do this. Next. And we put the GPS collar for the elephants. This is very good because every day, every 2 hours, we can monitor where the elephants are. The group of elephants, they make significant conflict with the local communities. And using the satellite, we can monitor where they, and then we can push back them the forest. these are very good point for the researcher, how, if you are willing, working for this analyzed data, because we not analyze data yet. We collect every day, but the data not yet analyzed. We need to invite you, the students or the researcher, to help us to analyze. Because like I said, I don't want to focus for the research. For the research, we need to invite another institution or another researcher to analyze this. This is free. Next. And we build the wildlife barrier system using the host, the snake fruit is very effective to mitigate the conflict with the elephant or another wildlife. Because snake fruit with the spin, and they don't like it. And also the snake fruit, the local communities can harvest. And this is good economic. And also we build the electric fence and trains and other thing. Yep. Next. This is also another important for the research. We manage 2 research stations now. And in this year, we will another 2 research stations. Some of the biologist students have been visit this and doing research and is regularly. And we hope many other students can doing research in here. And this is also for free. We never charge for the students. So if anyone doing a research in here, please care about your transport from Banda Aceh to the station. And after that, we're taking care of you. Your food, your facilities, room, and other thing. Just need explorations. And yes, another important, why we build these facilities for rhino breeding center. Even I don't like a breeding center because the wildlife can survive by themselves if we only care about protect their habitats. Because this is very expensive. You can imagine every year we have allocate around 300,000 US dollar for the one facilities. This is similar than 10 teams of the wildlife protection team. But for rhino, we have to, because in some area there is no breeding indication. They're still alive, but no breeding indication because might be of the sex ratio or the fragmentation, habitat fragmentation. So our planning to build the facilities in Aceh Timur on the progress norm, and later on we will capture some of the oceloted rhino and bring them to the these facilities. And we hope they can survive, they can breeding, and then later maybe after we have around the 15 individuals, we will release back to the forest, to their habitat with the very secure of the area. We have to be very careful about the rhino because in last few years, since 2019 until this year, we lost 26 Javan rhino in Ujung Kulon because of poaching. And this is half three of the last population of the Javan rhino. We don't want to happen in the Sumatran rhino in Leuser. Even we lost of the Sumatran rhino in many places in the world. The Sumatran rhino extinct in Malaysia, maybe only 1 or 2 in Waikambas, maybe less than 5 in Bukit Barisan Selatan, and maybe only 1 in Kalimantan. So this is really the last hope for the Sumatran rhino in the world. This is in Leuser ecosystem. Next. So this is not only our work. This is how we can build the collaboration between the NGOs, with the governments, with the police and other. We can see the forest loss, deforestation in Leuser in last few years stagnant around the 4,000 to 5,000 hectares yearly. Still big, but very difficult to reduce until zero in leisure because, as we know, we are in the tropical, very high rainfall, and then some natural landslide happen in the deep of forest. So we cannot control that. Next. Next, please. And this is the ratio of the snare in the leisure ecosystem after a few years working constantly, how we can reduce the number of snare per kilometer of the patrol team. So last year, there is only 0.01111 snare per kilometer. So to reduce this until zero, we need another patrol team. This is really effective and very useful how we can increase the population of the many species next. And today we can see this is so many young tiger in leisure. And if the local indigenous people knows about this, they're not happy because there's so many conflict with the tiger now in Aceh. And young individual, mostly, they're looking for a new territory, and unfortunately, they close to the local communities and then make a conflict. We can see the orangutans with the baby, they drink of they eat the soil because of the mineral. We call it salt lick. So, wildlife is clever. They are not stupid. They know what they need. And so sometimes they turn to the land and eat the soils for their mineral and back to the tree. So many baby elephant as well. And deer everywhere now. This is really one very importance of the conservation in the world. So conservation is easier. Next. So many species in here, like Sumatran goat, serum, deer, small cat, and Argus pigeon. You know, this is not in the natural forest. I mean, natural, but this is the palm oil area. Next, please. So this area we restored a few years ago, and a few years ago, 2 years ago, we identified the biodiversity in this area without any human intervention. 109 species of plants, 20 species of mammal, and 129 species of birds come back by themselves in this area. So this is very good for the research area. And back again, they come back by themselves. We don't need any human intervention in the nature. And some of the people said, why about the plant biodiversity? And based on our data, there is no dominant of the one species in this area. First 5 years, yes, dominant by the Macaranga. But after that, they're stable until today. After 14 years, 15 years, there is no dominant of the single species in the restoration area. Next. Look.
An informative presentation from, he tell us about the current situation, the biodiversity loss, and one key message that I actually take note from his presentation was that how protecting Aceh biodiversity is actually means protecting our economy. This is in fact very interesting because we used to destroy our forests for the sake of increasing the local economy. So I think the main key message is that we should be proud of our ecosystem. Our kampung ecosystem has a huge impact for the global interest, and we should work together with the conservationists, students, and researchers to research more about our ecosystem. Okay, thank you again for Pak Rudy for his presentation. Hang in there, we still have one more keynote for this session. Unfortunately, she will not be with us today, but she will be presented online. So Dr. Lindsay Benin is a senior statistical ecologist at UK Centre for Ecology and Hydrology. She has over 15 years of expertise in forest ecology, specializing in the impact of environmental change on forest structure, biomass, and carbon processes. Currently leading a major restoration initiative in Sumatra to the Darwin Initiative, she also heads for the Forester Network advancing evidence-based strategies for tropical forest restoration. Her innovative work, such as the Copot project in Ghana, has fostered collaboration among scientists, NGOs, and farming communities to promote climate-smart agriculture. Please welcome Dr. Lindsay F. Benin.
Lindsay Bannon from the UK Centre for Ecology and Hydrology in Edinburgh. Thank you for inviting me to speak.
Climate and people, and just talk about some of the project work we've been doing in the Southeast Asia region in general, and then also in. So, really, what I want to cover is really your perspective on forest restoration in the Southeast Asia region, and then how climate biodiversity and sustainable livelihoods all coming together under this theme of restoration. How we can deliver restoration on the ground and the factors that are important for consideration when we're doing restoration. And then look at some case studies from project work. And also I'll be giving some reflections on my recent trip to Colombia for the COP16 meeting on conventional biodiversity. So first, in large-scale perspectives. So we know in the
topics, there's been historic changes in land cover due to demands for other land uses. And for example, between 2002 and 2012, 6 million hectares of Indonesian primary forest was lost. And globally, we've seen these losses occurring, and the result of that is that 19% of remaining forests are within 100 metres of a forest edge. So we're seeing these big changes to the condition of these forests. So this has got people thinking about how can we deal with these challenges. And there's a lot of opportunity for recovery and restoration of tropical forests. So you can see from this figure from Cook Patton et al. In Nature in 2020, across the tropics, there's this great potential for above ground carbon sequestration in the tropical regions. And we have these broad scale targets through the UN Decade on Ecosystem Restoration and, for example, the Bonn Challenge to bring 350 million hectares of degraded and deforested land back into restoration by 2030. So we've got these large-scale aims and ambitions, and then we also have these country-level targets for restoration activities, and this might be by area or by number of trees. And so we need to think about how we can do restoration well and really optimize our chances of success and use our small resources efficiently to get the best outcomes possible. Now, with restoration projects, we generally have quite little information on what the outcome of these projects are. So a paper by Martin et al. in 2021 showed that quite few of the organisations involved in restoration are sharing information on monitoring and how tree survival, for example, is happening in restoration activities. So a few years ago, we initiated a project called Forestore, which is funded by the National Environment Research Council in the UK. And this was to bring together a network of people interested in restoration evidence and restoration practice. And I'd encourage anybody who's interested in joining this network to reach out. There's a slide at the end for contact details. So if you're working in that arena, please do join up with us. So we initiated this network so that we could start to bring together some of this evidence, which could then be used to feed into restoration practice. And the outcome of this work was that we compiled a large database of restoration sites where planted seedlings and saplings were monitored throughout the project. And it resulted in a compilation of 176 sites, and over 200,000 seedlings were monitored. And this slide just gives a bit of a brief summary on what that database looked like. So the map at the top was the sites where survival had been monitored, and then below that was where seedling growth had been monitored. And we compiled information around the number of species planted, the duration of the study, how many seedlings were monitored, and we included information on sites that had been planted on mineral soil and peat swamp forests as well. And then different habitat conditions. And at the bottom of this slide, you can just see the reference to this paper, so you can read up in a little bit more detail. So what did we find when we compiled all this information? I'm just going to show some fairly brief results from this paper because there's quite a lot in there. We found that on average, after 1 year of planting, 1 year after planting, 18% of the trees planted had died after the first year, and then after 5 years, on average, 44% of the planted trees had died. So we can see that we have this change over time, but what was really noticeable was that we had a really broad variation with it around those averages, so lots of variability in the data year to year, and a lot of that was accounted for by site. So some sites tended to have very high mortality and some had low mortality. And we accounted for broad-scale environmental factors, so climatic factors, and we also included some information about the nature of the plantings, so species richness and things like that. And very little of those factors explained much of this variation, but one particularly important one was this consistent difference in mortality, depending on whether there was tree cover present or not. So we found that on average the mortality was lower when there was still some remaining forest within the site or remaining trees within the forest site compared to more open sites, which tells us that there must be some benefit to be gained from having trees within the landscape where you're planting, and this could be microclimatic or other important factors such as soil conditions and symbiotic relationships. Other things we found with this study were that on average, in the first year after planting, the mortality was higher in the peat swamp forest than the forest on mineral soil, which indicates that this is a particularly challenging environment for restoration. We also found that the height of planting had a positive impact on survival. So larger trees were doing better. So stepping away from the individual trees, we also see that at the community level, there are benefits of restoration activities such as enrichment planting or other types of intervention, which could include climate cutting and this kind of thing. We can see that the above ground carbon accumulation is higher where we have some active restoration compared to natural regeneration. In this example, in Sabah in Malaysian Borneo. We also explored this within our synthesis work, and we found the evidence to be quite mixed amongst sites, and actually very few sites were looking at this community level. So it's a bit of a gap in the data and our knowledge. Another interesting aspect that we found with this data set was that in general, at the site level, the number of species planted was quite low. So the median number of species planted was 3 within our data set. Although across the data set more broadly, 625 species had been used across the plantings. So it shows that there's opportunity there, but on average, people are planting quite few species at any given site. And there's some work looking at why this might be the case. So why is planting in a diverse way quite challenging? It may be because there are singular objectives at the site, so more of a focus on, say, for example, carbon accumulation versus biodiversity. People engaged in restoration might be risk averse, so they don't want to throw lots of resources planting species that they don't know whether they're going to do well or not. There might be challenges around sourcing multiple species and poor seed supply networks and infrastructure, or a lack of capacity or capability to source from wild populations. There may be a lack of quality control for native species and a poor knowledge around nursery processes. And there may also be a lack of information around how species are going to respond to environmental conditions. So there may be a tendency to plant generalists that people know are going to do quite well. These are all really understandable reasons. But this significant opportunity for involving diversity in restoration in Southeast Asian forests. So a recent review by Antonelli et al. In 2023 highlighted that there are 14 of the 32 plant diversity dark spots in tropical Asia. So we're missing some knowledge around the plant communities in tropical Asia. And if we can gain some of that, it may enable us to bring more conservation into our restoration decision making. So just to highlight some of the key lessons learned from this aspect of the work, we found that restoration has potential, but survival rates are highly variable, and that means that there's risk associated to restoration. We can't simply trade off natural forests for thinking we can just restore them later, because we don't know what's going to happen. This may challenge land area targets in terms of our capability to restore large areas of land. Forest condition was a consistently important factor, so tree survival did better where there was existing trees in the landscape. And this tells us that conserving remnant trees in the landscape and forest patches could be really important to the success of restoration as well. We had this intersite variation in survival, and it's really interesting to think about whether we can harness information about successful sites to raise performance and strengthen capacity across more projects. So sharing that knowledge across projects. Whole community studies were rare, and so we need more information to more tightly pin down the approaches to restoration, whether active restoration is an important contribution. Restoration offers an opportunity to reintroduce lost plant species, but it requires new capability and capacity. And another thing that we discuss in the paper is the social context and indicators. These may be even more important for reasons for failures or setbacks. They might not just be environmental factors. So moving on slightly to actually the key topic of the conference, aligning climate mitigation, biodiversity and livelihood goals in restoration. Tropical forest restoration is considered one of the key ways that we can address the biodiversity and climate crises, whilst also supporting livelihoods and well-being of local communities. And to be able to meet these aims of the long term, we need to have effective restoration actions that are sustainable whilst also benefiting people, so that the ecosystems and the people are working in harmony. And this is sometimes referred to as the climate biodiversity livelihoods nexus, how these things all meet. And this is really what is at the heart of the Kunming-Montreal Global Biodiversity Framework. I think this is a really nice figure for easily taking in what the GBF, as it's known, is all about. These different aspects and targets to halt loss and restore nature, use lands and seas sustainably, share benefits and services, and mobilize necessary resources. When we're thinking about delivering forest restoration, we need to think about these different questions or aspects. Why are we conducting the restoration? Where will we do the restoration? How and who will be involved in that? And this really helps us get at the heart of the best approaches for restoration for a given landscape. The purposes and desired outcomes of restoration can vary, and it will depend on the stakeholders working in that landscape as well. So primary goals may be around biodiversity conservation, or it may be around economic income and the provision of food and fuel, for example, or medicinal products. It may be around sustainable timber production, or it may be about adhering to legal frameworks and mitigating other negative impacts. It might be around generating green finance or reducing erosion. So all of these different aspects can come into play when deciding what the key goals of restoration are. And there may be multiple objectives that can be ticked off in one go, for example, in a synergistic way, or they may be competing, they may trade off against each other. So let's take some of those previous examples that we were looking at. With the carbon capture, we may be thinking about wanting to achieve a rapid development of forest structure, and we may want to use well-known low-risk species that are abundantly available so that we can achieve good survival and growth rates. If we were targeting biodiversity conservation and enhancement, for example, we might think about a broader suite of species and encouraging natural regeneration to support that breadth of species. We may be wanting to attract dispersers and other wildlife and doing some spatial planning so that we can imagine some corridors where wildlife might move through the landscape. And to achieve synergies, you may think about how you can achieve more than one of these objectives at the same time. So I just wanted to share this example of some restoration activity happening in Sabah. So an organization there is working on the Kinabatangan River to try to support both rare plant species, but also support wildlife, support the capacity for wildlife within the landscape. So they're particularly focusing on actively planting certain plant species that would not come back on their own. And they've been thinking particularly about the dipterocarps and the important habitat that they provide for animals in the region. And they've also been thinking about introducing rare species and also fruit trees that would attract and support the local livelihoods. Restoration can take on different types. This is a scheme outlined by Disacco et al. In the 2021 paper in Global Change Biology. And they outline these different types of forest restoration. So the first one is around protecting native forest. The second one is around restoring native forest, and they really want to maximize rates of carbon sequestration and the recovery of biodiversity and ecosystem services. Then they outline livelihood native forest, which is about maximizing economic benefits to local communities while also trying to meet some of these local, regional, and global requirements for carbon sequestration, biodiversity, and supporting ecosystem services. And then they also refer to an agroforestry approach, which uses existing agricultural land and then provides a mix of benefits within the landscape by introducing perhaps more trees and reducing the pressure on native forests. And then they talk about protected native non-forest, so other ecosystems. And it can be quite useful to think about this framework in terms of different restoration and rehabilitation activities that may occur within a landscape. So within a landscape, forest landscape restoration takes on a mosaic of interventions. So it's not about just selecting one of those and having it as a blanket across the whole landscape. It's about choosing where these different interventions may be most appropriate for the stakeholders within that part of the landscape, and therefore meeting these demands of both the environment and people. And alongside this, when we're thinking about how we might go about restoration, there's also the fact of what intervention might be required dependent on the level of degradation or disturbance that's already occurred to what extent can recovery occur naturally and what requires interventions. And this is just quite a nice schematic that shows the different grades of intervention that may need to happen, depending on what the degradation and disturbance history is like in that part of the landscape. So now I would like to move on to sharing a bit more around case studies in Sumatra that we've been working on through a Darwin-funded project. And again, it's very much around this nexus of climate mitigation, biodiversity conservation, and human livelihoods. So the title of that project is Strengthening Community Capacity for Evidence-Based Restoration in Sumatra, Indonesia. And I'm really fortunate to have a great team of partners within this project. We're partnering in Sumatra with Fauna and Flora and KK Iwasi, and also Bryn. And we've got other partners in the UK, Plan Vivo and the University of Kent. And this project has four main streams of activity. So we've been looking at planning and prioritizing where restoration may happen. And then we have been implementing restoration in 2 case study landscapes. We've been developing ways for efficient restoration monitoring so that organizations involved can try to do this in the most efficient way possible, because it's a big use of resources to try to monitor these sites. And then we've been looking at income generation potential at these sites. So here are our 2 landscapes where we've been focusing, and topically for this conference, we've been working with partners to work in the Ulu Masin landscape in Aceh, in a Hutan Desa landscape, where there's some really nice quality forest, and then also some areas where the communities are using the land for rice paddies and mixed fruit tree crops, and also some grazing. And then we're also working in Bengkulu, which is a different social forestry scheme. It's Haka M and Hatta A. This is a coffee growing landscape where they've also been growing durian and some other crops. And there were some areas where they previously considered timber as well, but they've sort of changed their approach in the landscape. So it's really nice to have these 2 contrasting sites that we can apply some of our questions to. And at the heart of community-led restoration, we need to bear in mind local context, the free, prior, informed consent of people in those landscapes. And this, what is fundamental is the co-design aspect. So it's really being led by the communities, what they want to happen in their community and on their land. And the project is designed to be collaborative so that we offer support with the community. We recognise land rights, and it's a plan for the community to have autonomy over their sustainable land management. So I'll just talk through briefly some of the units of activity that we've been doing within this work. So we've been looking at participatory land management planning and multi-objective restoration prioritization, and we've been doing this in different scales. So we've been using some land classification at the site level so that communities can understand what the nature is of the land cover in their project area, and use that to inform how they want to manage their land going forwards. And then at the larger scale, we've been looking at how different sites might be prioritized in terms of restoration by taking into account biodiversity and carbon sequestration potential, and also poverty alleviation. So I'm going through all of this quite quickly, so do please feel free to reach out with any questions you have. So in terms of the participatory land management planning, we did some work around the mapping. So we, in collaboration with the team on the ground, conducted some ground checkpoints so that we knew what was there on the ground, and we combined this with satellite data from Planet Labs. And we used this to inform a land classification using the remotely sensed data. This involved combining lots of different data sources and seeing how those different data sources contributed to the improvement of the maps. And then ultimately, a land cover map was produced, and this was used to inform land management approaches in the landscape. And the community have been using this to decide and define protection zones where they want to try to retain good quality forest, and kind of make that commitment. So we've also been thinking a lot about how these rehabilitation and restoration activities within the landscape can also protect and enhance plant biodiversity in these mixed-use landscapes. So we wanted to amplify local knowledge, and we have been thinking about defining these protection zones, and also whether we might employ activities like assisted natural regeneration, which would enable regeneration of seedlings by suppressing weeds, for example. We've been thinking about ways that we might protect shade trees in the landscape, and then also where in the landscape native trees might be beneficial, maybe potentially as boundaries between land plots, as corridors or riparian buffers. And then also one approach is to set specific targets for native, rare, threatened and endangered species and make this part of the project. And we've also been talking to the communities about any culturally important species. And within the landscapes, they're talking about developing these community nurseries, which may feed into the green economy if there's demand for these seedlings beyond just the project site. So in this figure, you can see the nursery of the native trees that's been developed in the site at Aceh. And we really identified that it's important to compile lessons learned about propagation of these species and how to handle these native and wild species in a nursery context, because there may be quite good nursery information available for commonly planted and distributed seeds, but not necessarily seeds that are collected from the wild. So there's a big learning process. In Bengkulu, we've also been trying to understand what's important to the community, where there's culturally important species there, and they talk about the tanam tree, which is a species of dipterocarp. And they've been really wanting to expand the range of native MPTS species that they're using, which has economic benefits, but by diversifying crops, you can also create more pest resilient landscape. And there's also been knowledge sharing around understanding ecosystem benefits of shade trees, and also to improve productivity through agricultural approaches, so that the people are really able to get the most out of the land. and this will hopefully reduce pressures for further land clearance. Another aspect we've been considering is disaggregating goals according to different social groups. So it could be based on gender and age, so that different interests and perceived outcomes and benefits are accounted for within the planning of what happens in these project areas. And we really hope that this will enhance participation and buy-in across groups and encourage inclusion and equity. And we've also been mindful of respecting local customs and dynamics in doing this. We also hope to engender a view of adaptive management for long-term success. So it's not about simply having a plan at the outset and sticking to that. It's about continually evaluating and updating what's needed within the landscape and how things can be done better and continually improved. And this involves monitoring and evaluating and reassessing what's required. So it's been really interesting participating in the COP16 meeting and seeing the really important outcome for the new subsidiary group for Indigenous peoples and local communities to input into conservation and restoration strategy. It was a very lovely moment during the meeting when that particular negotiation was resolved. It's really important within these community-led restoration projects that this co-design is at the heart, and that locally relevant goals and priorities are at the heart of what's decided. But there's a lot of talk within the global biodiversity framework world about bioeconomy opportunities and how that would generate resource to local communities for conducting restoration and also generating income and livelihood benefits. So I think this is a really nice way of depicting our interactions with people around this work. So this is a really nice drawing by a child who submitted to a competition, and she talked about this drawing as representing the land that binds us. Humans are woven together. If someone gives up, others fall. We're all connected to our planet and to each other. And I feel really grateful to have been welcomed to conduct this work and support the work of others in Indonesia around restoration. So with that, I will say thank you for listening, and please do reach out with any questions. Many thanks to the funders, Natural Environment Research Council and Darwin Initiative, and also my collaborating partners. Thanks again.
So if any of the audience have one or two questions, please, you're welcome to ask. Anyone? Okay. Two? Okay, we got two. Can the committee there help with the microphone? Thank you very much.
Bismillahirrahmanirrahim. Thank you for the opportunity. My name is Sarah Salsabil. I'm from Biology Department. So my question is simple.
how can we make it balance between the economic beneficial and the sustainability of the ecosystem? Because as time passes, it is increasing the supplies, the demand of supplies by maybe by the surrounding communities or another companies. So that's all. Thank you.
Thank you very much. I think we get the next question, and then you can answer it all together. Next question, please.
Assalamualaikum warahmatullahi wabarakatuh. My name is Arnelita Yoziana Rizky Ramadhani. I'm junior high school in Tekunya Arif Fatih Bilingual School. So main question is, why the palm oil factory got the permission to use daily user area? As we know that this area is protected to Suaka Marga Satwa. Thank you.
Thank you very much. The first question was about balancing economic and protecting ecosystem. The second one was about why palm company use the forest, protected forest. Please, Pak Rudi, if you want to address the question.
Yeah, thank you. Very good questions. And yes, many years we're talking about the palm oil because the massive deforestation in the world by the palm oil. And we are the biggest producer for the palm oil. And so this is about the economic. I will combine between these two questions. Because of the demands, so 7 billion people know every day we need the food. And every food know our consumption contains the palm oil. And also a lot of another food production that we need for the everyday. And we cannot stop because this is we have to. But today, in last few years, I think in last 6 or 5 years, many companies changed. And previously, they only care about their supply, to secure their supply, and they don't care about the nature. And after many people in the world care and given the question to the companies, and today many big companies, they care about their supply chains. And so that's why we know about the certification, food certification. In the palm oil, there is RSPU, Roundtable Sociable Palm Oil. So this company know, many companies, sourcing only from the certified palm oil, and including in Aceh, no. So they don't want to sourcing the palm oil from the illegal, from the forest, from the conflict of land, from the including we call NDPE, non-deforestation, exploitation, and pit and exploitation. So deforestation means we cannot open the new land in the forest, and pit, especially in the pit swamp. Even we call it's only 1 meter deep. For the industry, no, they cannot allow from the pit swamp. And the third is about exploitation. Exploitation means this is human right, child labor, and another conflict social. They cannot use that. So this is mandate, no, and everyone care about this, especially the big companies like Unilever, PepsiCo, Nestle, Mars, and Protect and Gamble. And in near future, this for including for all of the production. So that's why in next year, the original plan, this is the 1st of January 2025, all of the product, there is 10 product cannot enter to the Europe if not clear and clean. But the latest news, they delay until 2026. So we can imagine, without the certification, we cannot sell our product to the Europe. This is terrible for the industry, but this is good for the environmental. We only producing from the responsible production here. And then today, we're working with the many farmers in Aceh and the companies. Only how we can increase the productivity. We don't need a new land, honestly, because so many abundant land in Aceh. There is nothing. There is our very low productivity. So our work now, and should be from this university as well, how we can, by the research, how we can increase the productivity with the current land. And today, in Aceh, only in 1 hectare of the palm oil, we only produce around the maximum 13 tons palm oil per year. This is very low compared to the Malaysia. Malaysia can produce around the 32, I think, tons palm oil per year. If we can increase the productivity, and that means this is 3 times of the current production, this is make our people will happier because with the bigger money and bigger impact on the economy. So, and then we don't need any expansion of the land. And then we don't need any new palm or the new plantation in our forest. So, the second question, back to the first question, is about economic. No, so a lot of potential economic from the forest. No, we talk about the carbon, we talk about the medicines and tourism, and in many countries, like Costa Rica, as we know, when they peace, there is no war in Costa Rica, the first plan from the government is not to build economic. Well, economic is important, but their strategy is planting the forest, protect the forest. So they're using the military and the police to protect the forest. And today, as we know, Costa Rica become the biggest market for the tourism. They get a lot of money from the tourism. This is benefit from the forest. And many other countries, we call ecotourism. This is growth very fast, and everywhere now, including in Bali, so many people, they love with the nature, and they spend a lot of money. When every year in Leusser, we invite, I think, around 100 or 200 visitors. This is special visitor. They are from the rich persons. This is not the mass tourism we will. But they spend a lot of money because they have to rent a car and then spend in the facilities, and we can sell it. Aceh has a very big potential for the ecotourism. We have many beautiful islands. We have the potential with the beautiful forest. And also, like in here, this is very close to Janto, to Selawa, and everywhere. This is potential. And if you travel from Banda Aceh to West Coast, to Tapak Tuan, we can see the beautiful of the land there, the beach, the oceans, and in Pulau Banyak, in Sabang. So this is our potential. How we cannot grow? Oh, we always talking about the Sharia law that's not allow the alcohol. And you know, right now like a Qatar, every year is very small country, but around the 35 million visitors every year visit to the Qatar. This is very big economy. They are Muslim, and also Saudi Arabian build the economy from the tourism now. as we know, many million of people visit to Saudi Arabia for the Hajj or for the Umrah. This is very big economic. And also, they also boil another facilities for the tourism now. It's very massive. And the most important for the future economic, this is from the ecotourism. This. So very big market. So we will know. We build the company, local company. This is for the how we can build economic from the non-extractive economy, from the tourism, from the carbon, from the biodiversity. We can do it. So there is no conflict. In near future, there is no conflict between economic and nature. And the second question about the palm oil. This is, honestly, this is because our fault. We consume a lot for the everyday. Some of us visit to the restaurants and we order so many food, and sometimes half of this is the waste. We not consume all. But the impact, if we lot of consumption, that means we need sourcing from the nature, like a rice, like a palm oil, and anything else. So if we can reduce our waste or our consumption, this is very impactful for the nature. And so that's why many local people or the companies expand their palm oil in the rawa singkil or the nature or the forest. First, because of the lack of the protection, forest protections, and also because of the increasing the demand. And very important for the governments to increase the protections, and also for the companies or the customer like us, we can ask the producer to only use from the responsible sourcing. They have to using this certified, and Indonesian also boil this own certificate. We call this ISPO, Indonesian Sustainable Palm Oil. And Malaysia also, they build the MSPU, Malaysian Sustainable Palm Oil, and many other countries also build the same thing. So please, we need to use only with the legal and then with the green productions. That will increase our economic and will save our nature.
Thank you very much. I think the keyword was being sustainable. And the challenge is how do we change people's mindset? Okay, I have a news that Dr. Lindsay is actually with us today, so she can actually answer your question if you have any. So does anyone have question for Dr. Lindsay? Everyone hungry, apparently. Do you have any question for Dr. Lindsay? We have one participant. Okay, please. I think due to our time constraint, we can only take one question for Dr. Lindsay.
Thank you for the opportunity. My name is Sarah Salsabil from Biology Department. My question is, before it is mentioned on the PowerPoint that the best tree that we can plant, for example, is like pohon durian, Duriozibotinus, and ficus. So my question is there any specific criteria or particular parameters to choose the most effective trees to prevent climate change? Thank you.
Thank you very much. Dr. Lindsay, you can answer the question.
Hi, everybody. Sorry, I think hopefully this works better. Sorry, I had an echo there. That's a really good question. So that example, the purpose of selecting those particular species was to provide fruits that would attract the local wildlife and support the wildlife. So in that case, that was one of the key examples, key motivations for the restoration. In other places, the motivations might be different, and people are working quite hard on developing methods to support species selection, and the objective might be around selecting species that are of conservation interest, for example, because they're threatened or they're rare or they're representative of a region. Or it might also be because in that area they're known to survive well and therefore can really contribute to the carbon sequestration. So there's different objectives for choosing species, but then there's also these new methodologies that are out there to support people with the species selection. And actually we have a new project about to start focusing on Kalimantan, but focusing on this exact question. How can we design restoration plantings that can support biodiversity conservation as well as climate mitigation and also could be resilient to future environments. I hope my sound was okay there for you. Sorry about that.
Okay, thank you very much, Dr. Lindsay. I think that's conclude our session. Thank you very much for your attention. And I think MC will take over the next things. Thank you again, Dr. Lindsay and Pak Rudy, for your insightful presentation. Assalamualaikum warahmatullahi wabarakatuh.
We kindly ask the invited speakers and the moderator to come to the stage for the presentation of mementos. For Dr. Lindsay F. Banin, the memento will be a certificate, and here is the certificate.
For Mr. Rudy Hadiansyah Putra, the memento will be presented by the head of Biology Department, Mathematics and Natural Sciences Universitas Syiah Kuala, Professor Dr. Leni Fitri. Now we invite Mr. Rudy Hadiansyah Putra and Professor Dr. Leni Fitri to the stage.
We also invite the moderator, Mrs. Elizar PhD, to come to the stage. Thank you to our invited speakers. Thank you to our invited speakers, Professor Dr. Leni Fitri and the moderator. We kindly ask everyone to return to their respective seats.
Dear guests, we have now arrived at the lunch break session. Feel free to enjoy the snacks and drinks that have been prepared for all the invited guests, presenters, and participants.
We also kindly remind all participants to keep the room clean by throwing the trash and the bins provided.
Please return to the room at 2 p.m. as we will continue with presentations by our keynote speakers.
Assalamualaikum warahmatullahi wabarakatuh. Kindly reminder to all participants, untuk konsumsi kami
Welcome back to ICBES 2024. Dear guests and participants of ICBES 2024, we kindly ask you to return to your respective positions as we will continue the event.
Before we start, we ask the presenters that will present their research topic to go to their own classroom, which will be guided by the liaison officer based on the room number list in the book of program. We remind again, we ask the presenter that will present their research topic to go to their own classroom, which will be guided by the liaison officers based on the room number list in the book of program. Thank you.
We now move on to presentation sessions by our keynote speakers, which will be moderated by Ms. Zaudatul Ulia.
Ms. Zaudatul Ulia is an industrial engineering lecturer and also a researcher of Arsiri Research Center, Universitas Syiah, Kuala. She is majoring in industrial engineering and specifying in supply chains and operations management.
We now invite the moderator to take the stage.
Assalamualaikum warahmatullahi wabarakatuh. Good day, ladies and gentlemen. Welcome to this parallel session of the first International Conference on Biology and Environmental Sciences 2024, proudly organized by the Biology Department, Faculty of Mathematics and Natural Science, Universitas Syiah, Kuala. Today's conference theme is Aligning Biodiversity and Climate Initiatives for a Sustainable Future, which highlights the urgent need to integrate biodiversity and climate solutions to create a more sustainable and resilient world. Today, we are very privileged to be joined by three distinguished speakers who will share their expertise virtually. They bring valuable insights from their research and initiatives across the globe from Malaysia, Taiwan, and Cameroon. Our first speaker is Associate Professor Dr. Muhammad Izzuddin Shakir Ishak from University Science Malaysia. Next, we have Chijen Ko from the Global Biodiversity Information Facility, or GBIF Regional Asia, Taiwan. Finally, we'll hear from Dr. Marius R. M. Ekwe from the Alliance of Biodiversity International and CIAT Cameroon. Okay, we are excited to gain knowledge from these outstanding experts. After each presentation, we will have a Q&A session, so don't miss this opportunity. As a reminder, each speaker has 30 minutes for the presentation. And once all the speakers present their material, we will have 15 minutes for discussion. Let us now begin with our first speaker, Associate Professor Dr. Muhammad Izzuddin Shakir Ishak from University Science Malaysia. He will be presenting on the topic Enhancing Earthworm-Friendly Ecosystems in Oil Palm Landscapes Through Livestock Integration Strategy, a Regenerative Agriculture Mission. I would like to read his CV. Prof. Shakir is currently a head or coordinator of Science at Professional Sustainability Program. He has education background for PhD for Earth Sciences from University of Ottawa, Master of Engineering Environmental from University Technology Malaysia, and BSc Environmental Science, University Kebangsaan Malaysia. He is a professional technologist, MBOT, and certified lead auditor, OSH 45000, and AMS 1401, Institute of Environmental Management and Assessment, IEMA. Please join me in welcoming Associate Professor Dr. Muhammad Izzuddin Shakir Ishak. Time and place is yours.
community. All right, and with that idea, they start to improve in machinery and engineering. Okay, and of course, this is actually the beginning of industrial revolution. And you can see the major invention during the first industrial revolution is the steam engines, where this actually used to power the factories to produce more products, okay, so that they can earn more profit. And of course, in the second industrial revolutions, at the late 18, the world is experiencing the mass production, where at this particular era, we can see that the internal combustion was invented to replace the steam engine. And we can see that more productions being produced, and unfortunately, we take for granted on our environmental management system. Okay, at this particular industrial revolution, we can see that the issues of the child labor, the issues of the hazardous waste disposal, and many more issues on the air pollutions, species extinctions, and whatnot. So these are the beginning of the environmental problems. Okay, but of course, the industries are more focusing on productivity. they start to look into how they can make their production more efficient. So they start to digitalize all their productivity. So make this era more focusing on the quality management in terms of how they can put all the system in a digital system. So that's why they evolved to the third industrial revolution, and then they evolved to the IoT, Internet of Things, where industry are more they are more lavish in terms of how they're gonna leverage all the resources by only within their desktop or their phone. Okay, the IoT. I mean, at this particular era, we can see that all are just within a single fingertip. Okay, but so all of this from Industrial Revolution 1 until now is more about how to make our productivity become efficient. Okay, and now we are entering the era of AI, where AI is being used to help us in making better decision. Okay, for our productivity. So AI can help us. So I think we are quite productive. Society now, with the evolutions of the IR industrial revolutions, right? But of course, if we just continue our business as usual, so means that we are more uh, exposed to the risk of natural resources exploitation. Okay, so in this case, of course, our nature is in in threat. Okay, and if we look currently, the data by United Nations, 40% of the global agricultural land is already affected by degradation. And if we look from the early agriculture revolutions to the IR 5.0, what I can say that we can divide the impact on the environment into two impacts. Okay, the first is the post IR 2.0 awareness. Okay, which more the awareness is more on the pollutions and waste disposal, and the second is the post-pandemic awareness. Okay, where people start to appreciate the impacts of climate change, GHG emission reduction. People start to notice the frequency of natural disaster. Industries start to put the climate risk into their financial consideration. And of course, at the end of the day, both impact are contributing to one issue, the land degradation. Our lands become scarce, less productivities, and of course, amid our growth population that is exponential. Of course, considering this situation, we need to reverse the damage caused by historic agriculture practices, as we looked before, which more focusing only on the productivity. And they currently now, they comes to the ideas of regenerative agriculture, right? Like what our invited speakers just now from UK also have emphasized the importance of restore, okay, our nature, right? And of course, if we look from our past studies, okay, my teams and also my colleagues, we did the studies on understanding the impact on the land degradations and water pollution. We can see that it's getting, worse, okay, from times to times if we don't do any mitigation measures. All right, and in the context of natural disasters in Malaysia, for instance, they have three components that contributes to this disaster. Which the first is the lack of the ecosystem services from forests, riparian zones, as well as the river. Okay, and of course, to restore this, it will need certain times, Okay, it takes time, energy, resources, and also the cost. That's why when the barriers, we can say that the natural barriers like forest, riparian zone, and also we cannot control, I mean, the sedimentation in the river. So this make the ecosystem weak and prone to any uh, risk of natural phenomena like unprecedented high amount of rainfalls. You can see that it's a very sensitive to the changes in the amount of rainfalls. So this is what we are experiencing right now, and that's why the awareness came on the climate change came after post pandemic. And considering the United Nations climate action, okay, they have 6 climate related action, but in this context, we're going to focus on the green transitions and also the effort to confront all the climate risks. All right. So these are the components that our research motivations are moving right now. And if you look from the soil as a resource, okay, it's actually the central of the biggest issues right now, climate change and the food security, because of soil also play their roles as the largest terrestrial organic carbon reservoir. So that's why this is one of the very critical resources that we need to address, okay, to put it as a priority. And of course, to determine the sink and source is actually the earthworm abundance. They play a very significant role in determining this sink or source of the carbon cycles in soil. All right. And how? Because of they can build or create the aggregates, okay, through their feedings and casting activities, so that they can stabilize the carbon trap within the aggregate. And furthermore, the humifications of organic matters by earthworm provide also a biochemical mechanism to stabilize the soil carbon. In other words, we can say that earthworm actually play a very critical role in regulating the carbon cycles in the soil system. So if we look from soil ecosystem services perspective in terms of regulating, okay, if in terms of the carbon regulation, so you can see that the casting activities and decompositions of organic residues is on our highlight, okay, and that is much on our studies, and you can see that fortunately what triggers the earthworm abundance is the organic input. Okay, so we can have this input by introducing the livestock integration practices, so that by introducing the livestock integration practices, they can intensify this organic input into the soil system, and with this condition, it will attract more earthworm or increase the earthworm abundance. Okay, our latest studies in terms of enhancing the earthworm-friendly ecosystem, we found that the earthworm, they favor, okay, the undergrowth and canopy. Okay, considering this undergrowth and canopy means that they need the understory vegetation protection, so that the soil is actually conserved by this understory. So with this healthy soil, they can also attract more macrofauna abundance and diversity, as well as play their functions to regulate carbon dynamics within the system. And this is, of course, the opportunities for us to reduce the chemical input, okay, from herbicide in this case, so that it can reduce the risk of the understory damage, okay, and also the risk of pollution. So by introducing the livestock integration, you can also look in terms of how it also can, in the long run, can support the food security agenda, employment of the small farmers, as well as additional income. Okay, so this is how we optimizing our land by introducing the biological input through livestock integration, and also restoring back our soil with the organic matter. That's the core. That is the strategies for us to increase the input. So you can see here how you can see that the difference when we use the cattle, all right, to maintain the weeding. So you can see it's a very neat and clean uh, uh, maintenance there. You can see after grazing and before grazing. So the understory is and the grass is well maintained by the cattle. So we no longer use herbicide for this. Maybe we reduce the herbicide. We can use at only at a particular areas only, not a blanket application, right? So and you can see that the difference between the weeding with herbicide applications and weeding with the cattle integration. So of course, with the herbicide application, the risk of damage understory is high, and of course, it also will lead to other environmental issues like erosions and whatnot. Okay, but when you have this cattle as our agent to weeding and also through grazings, okay, and of course this also as an agent for biological input. We tried to understand this further by looking or by doing our observation on the full herbicides plot versus free range, which are partial herbicides, and systematic practices to see how our soil ecosystem services works. All right. And of course, we know that in terms of the soil ecosystem services principles, they have principles of supporting. So in this case, we want to look at how much the SOM can be enhanced through all these practices, and in terms of provisioning nutrients, okay, this where we want to observe how much cast is being generated by earthworm, and also regulating the carbon storage. So the potential of soil ecosystem services to regulate this carbon. Okay, so this is how we design our studies to propose or to promote the regenerative agriculture approach. All right, and these are some results, okay, where I gonna show you in a figure, so that's much easier for us to understand. So you can see that the green is the systematic with the systematic livestock system, and the red is the free range, where we just allow the cattle to go freely within the oil palm plantations, and the blue is fully without any introduction of cattle practices. And you can see that the difference, okay, of the soil organic matters compare okay, is between 20 to 4%. Okay, you see that the plot one, which fully herbicides, only have about 4% to 10%, okay, of the SOM, while you can see that the systematic cattle grazing areas has more richer SOM. Okay, so if you look from in terms of the pH also is increasing with the enrichment of the SOM. same, the soil bulk density also decreasing with increasing the SOM, so you can have a very fertile and loose soil for plants, for plant nutrient cycles and whatnot. And from the principal component analysis, you can see that plot 3 is much more attributed to the organic carbon, all right, which actually plot 3 is represented by the systematic livestock integration practices, where the nitrogens and phosphorus is quite evenly distributed. But if you look into plot 1 and 2, they are only more on the TN and TP, which is, we can say that the dependence on the on the chemical input is much pronounced in plot 1 and plot 2. So this also in agreement with our data on the earthworm abundance. You can see that the circles, okay, the red circles is actually the systematic grazing practices. You can see even it can have more compared to the free range and also herbicide practices. All right, across the profile, okay, 0 to 10, even 10 to 20, and also 20 to 30 cm, you can find all across these soil profiles of the earthworm abundance. You can see more significance compared to the free range and also the herbicide. And in terms of cars, also you can see the productions of cars is much higher, all right, compared to chemicals, all right. And you can see here the cars at A, okay, the A, which is systematic practice of livestock practice, is about 8,000 to 1,000 cars per square area, okay. Compared to the free range is about 400 plus per square meter, and the herbicide, okay, the areas, the plot without the biological input from livestock integration is just about 60 cars. So it shows a very significant difference between across these plots, okay. And even we do the cluster analysis, we can see that the between the systematic, okay, marked as a blue, herbicide, green, and free range, red, you can see that the link is very strong there. You can see that 1 is very strong, 0 to -1. So you can see that the link between the class adults, okay, the cars production, the abundance across the profiles, they are very consistent in systematic, followed by free range, which is not that consistent, and then and then the free range, I mean the herbicides is much less, okay. It's just about 0 to -1. So the data is very consistent. And of course, different management practices such as livestock integration determine the earthworm community structures. The earthworm abundance and activities influence soil structures, okay, like pH and also the SOM just now. And of course, a shift in these practices may lead also to the change of the organic matters and pH, potentially alter the abundance and activity levels of earthworm, and what is the worrisome is impacting the soil resilience. And even if we look from the economic point of view, you can, if we consider the concept of the externalities, you can see that the economic valuation here, the external cost is quite high, okay. If you look at the blue areas here, at the left hand side, which is of course these are the costs that we need to pay, but it's not being reflected in market price. So that's why I mean we keep wondering why our farmers keep doing better and better, but they are suffering with the very high cost of of current, market, because of we don't factor in our ecosystem services cost into this. All right. So that's why this is a very important for us to reflect and revisit the way we do agriculture for the sustainable future. All right, we look at the regenerative agriculture missions. Of course, the ultimate is to increase the resilience of the soil system. If we refer to the Royal Agricultural Society of England, regenerative farming means the farming practices with improve the environment and with the soil functions taking a key role. So we need to make sure that our soil is not dead, our soil is livable for all microorganisms so that they can functioning well and of course improve our ecosystem services. So if we recap our discussion or presentation just now, the most important thing is to, when we talk about the regenerative agriculture, it's about how we gonna minimize the soil disturbance, how we can improve the soil covers, and how we gonna strategize the integrations of livestock. Minimizing soil disturbance means that minimizing the chemical input. Okay. Increasing the soil covers means that we need to conserve more understory vegetations in the landscape, agricultural landscape, and strategizing the livestock integration is important for us to increase the biological input so that we can increase the resilience of our soil system, mitigate the climate risks, and of course, the most important thing, to face the challenges of food security in future. So, strategically, we need to reduce the chemical input and find way to increase the biological input and conserve our understory vegetation so that we can so that is the way in terms of how we keep track on our sustainable practices. And of course, now we just look at the climate change as our biggest issues, but sooner or later we're going to face another issues of biodiversity collapse. So that's why we need to start from now to revisit our current practices, okay, by limiting our chemical input to our ecosystem so that we can, we can face the challenges ahead. Just to give you a perspective, if you look back at the Earth, the World Environment Day since 1970 to 2024, like what you can see in 1970s, they are more worried about carrying capacity of our Earth. And then 10 years later, they start to look at our resource scarcity. They realize that our resource is scarce, is not finite, okay? And then when it comes to 1990, they start to look into the impacts of climate change, okay, the awareness. And then comes to 2024, they start to know that our land is our future. But what I want to emphasize here, it takes about 30 years for us to appreciate or to aware that climate change is real. And in the context of Malaysia, we start to put that ESG compliance is important for all the, multinational companies to report, okay, their activities in terms of their GHG reductions, their environmental protections, socials, and also their governance into so that this gonna be already being compulsory to in the context of Malaysia. So we are now at this stage, and of course, regenerative agriculture is very important. Okay, we can now have ESG become our platform now to report our practices. So as I can say, a regenerative agriculture approach goes beyond traditional farming. by aiming to restore soil health, improve biodiversity, and build long-term ecosystem sustainability. In the context of this study, it was evidence that the plot with systematic livestock integration practice demonstrates significant earthworm abundance, consistent with soil organic matter content and soil physical structures, which are important indicator of the soil system resilience. And in the long run, resilient soil systems support sustainable agriculture practices and help mitigate climate change impact by sequestering carbon and preserving biodiversity. So those days, our focus only on yield, okay, how to increase our productivity. But today, we also need to look in terms of the intrinsic worth of the soil ecosystem entity, like the earthworm. Okay, so very critical, and also the soil conservation, so that we can enrich more SOM as insurance for resilience to environmental change in future. So I think that's all for my talk. Thank you very much, and I hand back to our moderator. Thank you.
Please give a hand claps for Dr. Shakir from Universitas Malaysia. Thank you, Dr. Shakir, for that insightful presentation on enhancing ecosystems in oil palm landscapes through regenerative agriculture. I know that there are a lot of questions coming up in our head right now, but before we are going to the question and answer session, I would like to move on to our second speaker, Chijen Ko from the Global Biodiversity Information Facility, or GBIF, from Regional Asia in Taiwan. Chijen will discuss about use and contribute to open data through GBIF for a deeper understanding of global biodiversity. I would like to introduce Chijen for a moment. Chijen Ko is a consultant for biodiversity data management. He is an enthusiastic biodiversity fair data champion inspired by the informatics for natural history collections. 15 years of experience across engagement, training, documentation, and web development within GBIF communities and beyond, seeking to repatriate expertise, strengthen skills, and facilitate data publishing and its use with Asian peers through active participation. His education background is in MSc in Biological Sciences from National Sun Yat-sen University, Kaohsiung, Taiwan, and BSc in Biological Sciences from National Sun Yat-sen University, Kaohsiung, Taiwan. Please join me in welcoming Chijen Ko. Time and place is yours.
Thank you so much for having me. Thank you, the moderator, for the introduction. I'm excited to share with you about GBIF and how to use and contribute to the open biodiversity data. I would like to begin my presentation with 3 research findings about our biodiversity related to climate change. First, cashew cropping. It plays a role in agriculture in West African countries like Togo. It's originally introduced from Latin America, but becomes more important in West Africa. Scientists want to examine whether it's sustainable to face, or when it faces climate change, whether the cropping ranges will be changed. So they ran a model for the species distribution with variables including soil, isothermality, temperature, seasonality, and precipitation. It turns out that cashews are relatively resilient to climate change, with the projection for the year 2055 predicting an 8 to 10% increase in suitable habitats. This is a good news for Togolese farmers. The second one, urban forests help regulate heat and provide shade and a connection to nature for many of us living in cities. In this study, researchers analyzed the potential impacts of future climate change on over 3,000 tree and shrub species in 164 cities across 78 countries based on the global urban tree inventory. Their analysis predicted that the highest exposure to climate change is in Helsinki, which is in Finland, Winnipeg in Canada, and Minneapolis in USA. Overall, up to 65% of species were currently exceeding their margins. The authors estimate that by 2050, up to 66% of all urban trees and shrubs will be at risk. And the third one that is about bats. We all have been through the pandemic. As bats are the host of dozens of SARS-like viruses, understanding their distribution is critical for conservation and public health. In this study, 35 bat species were modeled. The projection results suggest most species will likely lose their suitable habitats, which might lead to fragmented populations or bring more bat species closer to human populations. These findings about the effects of the climate change on our biodiversity have one method in common. They all use open data published through GBIF. And so do all the thousands of peer-reviewed publications. This figure shows the number of publications year by year since we started the literature tracking program in 2008. Now, on the average, there are nearly 6 papers published every day using the GP-mediated data. So what is GBIF? GBIF was born out of a science forum in OECD in 1999 and established in 2001 because countries recognize that tackling, understanding the issues about biodiversity cannot rely on any single country. Countries have to work together to share data. Therefore, they can have a broader picture of how biodiversity interact with the industrial and all aspects of human activities. So countries have to work together on that. So they put out a budget, invest, and to create this GBIF as a global biodiversity information facility and to promote open sharing, open access to biodiversity data, and also establish an infrastructure for flowing this data in order to for people to use. So this is the brief view of the infrastructure right now. And if we look at the right side, those papers that we are tracking can be roughly categorized in several major disciplines, which include ecology, evolution, and the 3 climate exchange examples that I just show you: human health, invasive species, conservation, food security, etc. And the scientific research findings using the data are also being used in the reports of global initiative like CBD, IPBES, IPCC, WHO, and FAO. The reason why scientists have this data to use to do their analysis and come to this conclusion because they are all supported by the infrastructure built by GBIF. So if you have gone to gbif.org, you know that you can put in species name and find and download the data that you are interested in, and then use the data in your analytical workflow. So right now, as the slide shows, that in the center is the GBIF Global Index. It comes from the data that is published by data publishers across the globe. And on top of this index, there are several services that can utilize these data. And these data comes from museums, natural history collections, and agencies and ministries. They conduct periodic surveys, for example, in national protected area. And then also field researchers, they pack their data in a standardized format and publish it. And the recent years that a large amount comes from the citizen science, bird watchers from eBirds, which is a larger data set in GBIF at the moment. We are having more and more business sector engagements. So before their construction or the business plan. They usually, if they use some kind, they need to use habitat. They will do a habitat environmental survey or analysis. Those data are also being shared through GBIT network. So essentially, these data include from the museums, it will be specimens, and there are also observations, literatures that are, for example, taxonomists that they write monograph describe species and referring to voucher specimens in museums. They come from literature. We also have a lot of remote sensing data. And right now, for people, for places that people cannot go to, eDNA is also becoming a trendy, and many people start to use like this method, also share eDNA sequence information into GBIT network. And these data are organized according to standard, and they are shared through GBIT under public licenses like CC BY or CC BY NC or CC 0. So they are open licenses and then allow for reuse of these data in a very diverse situation. Currently, there are 180.7 million records that are taxonomically identified with images, but there are more than 3 billion records on GBIT.org. For those multimedia information, they are not directly hosted by GBIT. They are provided by data publishers that have these multimedia files stored online somewhere, and then GBIT merely have the URL to retrieve them. So if you go to gbit.org, you look for species, like for example, in this example, Wafflesia, you can easily see that it will quickly give you a synopsis of the species information on gbit.org, including images. And if the data, most of the data have the coordinates, and they will plot them on the map, and from which data sets these data come from. You can find there's a download button, and when you click that, it will take you to provide you several options to download. It's a simple table or a format, a standard format we call Darwin Core Archive. And then if you click that, you need to understand that because you don't need to pay anything for downloaded data, but these data are available because of the efforts of all the data publishers across the globe. And so you need to understand why it's free for you to use. And you click understand, and then it will start preparing the download for you. You need an account to download that, and when the download is ready, you have this download icon ready to click. And don't forget, when you use the download data for analysis and publish your paper, remember to use the citation string that show in the screen in your as your references. And we so far can track the data use of GPU data because of people are citing that. And also because of this, people who contribute and publish data through GBIF also get credit, because as soon as you publish data to GBIF, and as soon as the data is being downloaded for someone else's to use, and data publishers can see that how many downloads of their data has been produced, and then when there is a paper citing your data, the publisher also knows. So this is a sort of credit feedback mechanism that is available on this global platform. So the data looks like this, and it's in the standard format. The data can be, I mentioned several times that because of the standard, the data can be shared. The standard we use is Darwin Core, which is not really a rocket science. It's basically your everyday terms. I believe people in our field, all of you in the audience knows that the individual, what does it mean by, for example, rights holder, what does it mean by collection code, what does it mean by basis of records. There are some terms that are organized following the standard, and we agreed that we use the standard to communicate with each other about sharing our biodiversity data. So globally, everyone follow the same way of sharing data, and therefore the data can be accumulated and then used in the larger scale. So this is how data get to be available in gbif.org and for people to use, and not only the website and the technical infrastructure, but there's also people, countries joining gbif and promoting the standards, helping people to use the standards, and running workshops and get people excited about and understand the benefit of open access to biodiversity data. And therefore, we have these many information available for research use. There's recent years that GP has put a focus on collection information. They developed this scientific collections.gp.org. We have this specifically for natural history collections users, because most of the GP data, when they aggregate as whole, have been used. They provide most benefit for those running larger scale analysis, species modeling. And then there's a significant contribution from the natural history collection community that GBIF has been seek to repatriate their contribution. So this portal is specifically built to allow people working in the science collection background to be able to focus on the collection management and exchange of information. The specimens stored in the museum and herbarium is particularly important because they are physical evidence of our biodiversity across space and time. So this is a way also to highlight the importance of these information. So if you are coming from the natural history collection background, these websites, as soon as you publish data using the standard on the GR Cycle websites, you can quickly get a lot of meaningful information from your data set and be able to present that to your colleague or for people to be able to find what do you have in your museum. So you don't need to, this is why it's beneficial to have a shared common infrastructure, because distribution on internet is free, and each of the participating museums, if they can use this global infrastructure, they don't really need to spend extra money to build their own websites, because once you build a website, the web technology evolves, and after a period of time, because of various reasons, like for example, security issues, your website cannot be can no longer be online, or you need to hire another another way for development with extra money for expensive engineers to revise your website and run the whole exercise again. But GP has countries already paid to build this infrastructure, and then therefore data publishers only need to focus on getting data following the standard and publish it. So mostly on the content part that they are most comfortable with. So this is a way to share the cost of distribute and use biodiversity data. This is more content that can be shared through the scientific collection websites. Many people will ask that, how do I get my data to GBIF? Can I, as an individual, publish data to GBIF? The answer is not as an individual. as an individual, you have to be affiliated with the organization because as a data publisher, the organization carries their responsibility to keep the data maintained online. Therefore, there's an endorsement process when your organization wants to publish data to GBIF. There's a form online, gbif.org, becoming a publisher, and in there you can find whether your organization is already registered or endorsed as the GBIF data publisher. I always recommend people who are interested in looking how to get data prepared, start it from the GP webpage, on the how-to click Start Guide. There you can see that GBIF currently supports what's the standard and supports 4 kinds of data sets, which is you can publish checklist of species, you can publish occurrence data sets, which can be observations or collections. There are also data quality requirements, although the standards itself doesn't require you to publish any, make any columns mandatory. But as an aggregator like GBIF, it does need some critical elements in order to allow people to find your data. So scientific name, of course, and the time of the event, and the locality. And with a better quality, you also are encouraged to provide your coordinates of the record. So these are aspects that you need to learn about before you start to think about how you're going to publish data to GBIF. There are tools online, like for example, we have a spreadsheet, a template. You can download a template and start to see that your data with you, whether how suitable they are or how ready they are to put into the sheet and start your publishing process. We have a biodiversity data mobilization course. As a part of the Asia regional support team, we run the course right now every year during spring and summertime. And the course is about 5 days of online sessions, which walk people through all aspects of getting data cleaned and improve the quality of data, and also how to get data published. And the IPT is GBIF produced that would allow data publishers to host their own servers to serve the data and publish data, which is also part of the course that we are running. So once the data is published, it's the software engineering and web development to process data, make global index, come to this page as what I want to remind you, and therefore you have the website to use. If you download Darwin Core Archive, and then it's the data looks like this. This is a checklist data set. You can see that this is the checklist, the core text, and it contains kingdom, phylum, class, order, family, genus, specific epithet, and for specific epithet, etc. And then you can also include description and also the molecular name. And this is Taiwanese data set, and there's a Taiwanese molecular name, and language is specified here as well. We teach people how to use data in a course. We introduce R. The R environment is much easier if you are dealing with a large amount of data. So in our environment with the RGIP library, it can help you interact with the GBIF API. So you can download the data and examine the data right in the R Studio environment. And our course also demonstrates how to do that. So as I mentioned, it's not only the infrastructure, it's also people. And we in Asia, we are running these activities to engage with people, to get people to know each other and also know more about GBIF, also know how to solve their questions about biodiversity data. So we every Thursday afternoon we have an office hour. For us, Vijay, Lily, Melissa, and me, we're online there for 1 hour, and people can just pop in for any questions about what they do, or they can also just come and chat. So it's really just a very casual time. We run workshops, and other than these activities, we also help looking at the GR cycle entries in the museums, and invite them if they are more or less ready to share their data, or just to let them know that there's a place that they can expose themselves. So what we do as an Asia Regional Support Team pretty much covers the whole course of getting data from raw data and getting data to standardize, cleaning, and validate whether they meet the standard and can be indexed by GBIF and publishing. We also help people to publish data paper, and as well as introduce skills like I mentioned, our studio to start their data use journey. GBIF is also offering a data camp license. So if you are interested in learning more technical informatics skills to utilize GBIF data, we have a data camp license for you to learn. One of the rewarding experience with the Asia Office Hour is that when people come here weekly and we have solved their questions little by little, they eventually will be able to get their data published. And there are also many questions that we answer frequently. For example, how to share measurements in a sample event data set, or if I have sequence data, how do I get it published? After this presentation, if you are interested, you are always welcome to come and access. Sorry, I just jumped into because of time. We are also happy to see many data sets published immediately after our training event. So for the training event in Asia, and for the last few years, we are happy to contribute, with the data publisher join our workshop, contribute more data that is published immediately after the workshop. So people can learn in the workshop and immediately prepare the data and published, which is quite happy. I'm quite happy to see the outcome. There are now many data publishing institutions in GBIF network. There are more than 2,000, and these data are very important because to conserve our biodiversity, we need data to make informed decisions and to guide our actions. Many of them, the data publishers actually are not from our region, Asia. Right now, on gbif.org, there are many data about Asia are actually repatriated from researchers and institutions outside Asia. So if we look at the participation countries map in GBIF, Asia is relatively empty now. It's critical for countries to step in and support biodiversity data management at the national level. I believe in Asia there are still tons of the data sets that are locked either in researchers' hard disk or not being standardized. There may be danger that if they're retired and no one look at the hard drive and the data will be forever lost. And those data are really valuable because it's the biodiversity they have been through. And thanks to our previous presenter, I couldn't recall him actually right away, Syiah. beginning by introduce our industry evolution. And right now we are at the beginning of exponential growth by AI. But please note that for AI to assist us with biodiversity, we need comprehensive data of good quality. So especially data from local publishers has significant importance because you can imagine that the data or the AI models that are trained by data from other places may not always have provided best fit for local use. So we need to first get data out. In order for the trained AI model to fit for our use. So I hope this presentation, as introduction to the audience here today, can lead to a new initiative to engage Indonesian academies and the public about open access to biodiversity data. So please help advertise and advocate this global mission. That would be my presentation today, and I'm open for questions. Please write to me after this if you have any questions. Thank you.
Thank you so much, Chijen. Please give a big applause to Chijen for a very enlightening presentation on the power of open data for biodiversity. Your insights on GBIF's contributions are invaluable for researchers and practitioners working to protect and understand our planet's diverse species. I know that there are many questions coming up in our mind right now, but we are going to come to our last speakers first before the question and answer session. We turn to Dr. Marius R.M. Ekwe from the Alliance of Biodiversity International and CIAT in Cameroon. Dr. Ekwe will present on mapping the vulnerability of priority native tree species in Central Africa to guide their conservation and uses in landscape restoration. I would like to introduce Dr. Ekwe first. He is currently a scientist, Tree Biodiversity for Resilient Landscapes Alliance for Biodiversity International and CIAT. His education background was Doctor of Philosophy for Forest Genetics and Master's degree Tropical and International Forestry from George August Universität Göttingen, Germany, an engineer's degree from Natural Resource Management from University of Abumi, Kalevi. Please welcome Dr. Marius R.M. Ekwe. Time is yours.
Today, I want to talk to you about the work we are doing in our research group to map the vulnerability of priority native tree species to guide their conservation and use in landscape restoration. This is the outline of my presentation. So I will talk briefly about the Alliance, and then I will talk to you about the tree diversity database we are working on, present a tool called Diversity for Restoration, and then I will present a platform called My Farm Tree before concluding. So for those who do not know the Alliance, It is actually the merging of two CG research center, one called Biodiversity International and the second one called SIAT. And we merged since 2020, so we are working like the same institution, dealing with a lot of research theme, going from food, environment, and consumer behavior, going through multifunctional landscape, climate action, agrobiodiversity, and also digital inclusion. And obviously, we are looking at the role of gender, youth, and social inclusion. So we are a global research institute working at the intersection of agriculture, nutrition, and environment. And we are working across the globe. So in Asia, we have our regional offices based in Malaysia, and with office in different countries. So the tree diversity database, it is actually a spatially explicit priority setting tool to guide the conservation and restoration. And why are we doing that? This graph show you the diversity of tree species we have in the world. And Africa has quite a lot of tree species, but most of them are also threatening, by climate change and different threats. And while they are also providing, a lot of product and ecosystem services important for rural population and also for the development of country in the region, especially in the Congo Basin. So the work I want to present to you today is something that we have initiated in Central Africa. So starting from Cameroon and looking at the presence of those species in all of the neighboring countries. So we prioritize 100 native tree species which are important, for the local population, which means they are providing to them different products and ecosystem services, but also species that are threatening and also important for conservation. And the objective was to map the vulnerability of those tree species vis-à-vis of climate change, but also other threats such as fire, conversion of habitats, over-exploitation, overgrazing. And we combine those the information generated to provide species-specific map to guide conservation and restoration action. So the methodology that we follow was like in 5 steps. The first step is to do distribution modeling using occurrence point of those species and also different environmental variables, what we call ensemble distribution modeling and the mass center, etc. So after the species distribution modeling, we also look at the different threats. I talk about fire, overexploitation, overgrazing, habitat conversion, and there are global available database, about those threats that you see in different sources. Like fire is coming from the so-called the MODIS active fire data, and we use those information also again to model those threats to each tree species in the region. And by doing this, we calculate, the species distribution. We also have what we call those exposure map. We look at the intensity of the threats, which go from zero, low, medium, high to very high. So if the color is very red, it means that those areas where the species occur are highly threatening. And then last, we do the projection with the future climate scenario RCP 4.5, so the scenario from IPCC. And next, we do what we call a threat-based sensitivity per species. And why? Each tree species have different sensitivity. When we take like fire, obviously, when there is a fire in a specific habitat, you will see at the end that some tree species are completely burned down and destroyed, but there are other species who can resist the fire and even if be able to germinate very easily. So we look then at specific threat data that really can help those tree species, to support the fire. And in this case, you notice that the tree species who can withstand fire have usually a very thick bark or they are able to raise fruit very easily. We combine all of those different threats and we go through the literature, herbarium, so to take for each tree species, we map, what are how resistant they are to all those different threats. So going from low, medium to very high. And we all of those information to create, this is the fourth step, to create what we call vulnerability map, which is actually, combination of the exposure and the sensitivity. And we combine all of those information together to generate then what we call the vulnerability map for the species. And with those map, it support us, to draw conclusion to determine like the particular area can be good for in situ conservation of tree population, where we can prioritize seed collection for future planting in other area, or also for ex situ conservation of seed sources in area with high climate change vulnerability. So we need really to conserve them, or also we can also promote active planting or assist natural regeneration in area under current vulnerability threat but low climate change vulnerability. So that's, and this is like a combined map, of all vulnerability for different tree species on the left, and also we combine all of the threats here. And then if you can see very clearly here, you see the species that are most threatened here is like a Prunus africana, which is a species highly used in Central Africa. It can treat prostate cancer. So naturally, there is a very big market, and those species are really threatening and on the list of CITES. So that's the work I present, is very important really to know the diversity we have there, to see how threatening are this diversity and what are the measures, to conserve them either in situ, ex situ, or even to promote active planting. So we really need to safeguard the diversity. And, we go further with this tool we call diversity for restoration, because it's good to conserve, but we also need to restore those materials. And basically, what this tool does is really helping you when you are in a particular location and you want to do tree planting. The tool can help you to select the best tree species which are suitable for this area. And then for each tree species, it helps you to select, the good seed sources and also give to you some information about how to propagate. So this is basically the way it works. So it's what I just said. The tool is really good at supporting decision making to choose the right tree species and seed sources. And it's a web-based tool where you will need to enter the tool and add the objective that you want, to pursue for your tree planting. And then you describe, the condition of the planting site, and you describe, the how is the condition, and also you add, what the future condition in those area. If you want like to plant a tree and the product that the tree will give to you, you want to harvest them in 20 years from now, then, for the climate change scenario from IPCC, you probably want to take a scenario that will take you to 2050, etc. And then the tool will run you this kind of result. So It's what I said, before. You can just go to a map and point to the location, and then you have the coordinate automatically, or you can also write it by hand, and then you run the tool. So we actually have the tool for different countries in Latin America, Africa, and also in Asia. We have it in Malaysia, Vietnam, and it's also being developed now for Indonesia. And those are the kind of results that the tool give you. So you see this option, the tool gave to you like three different options. If you want to maximize diversity and species suitability, the tool gave to you a list of 3 species that are suited for this particular area. It also provides to you a list of species if you want to maximize only diversity, or if another area you come to plant there. So you have this kind of recommendation here. It's telling you here to collect 75% of your seed from the seed transfer zone 1 and 25% from the seed transfer zone 2. And down here is, even if I advising you to collect a seed from 3 different zones to be sure that those materials are genetically diverse and climate resilient and will behave well in the place where you want to plant them. We also provide, like a sheet, maybe 2 or 3 page, to explain to people how to propagate those native species. So this is then the second tool that I present for more restoration. There is a new tool that we have developed recently, and then we are expanding now. It's called My Farm Tree. And My Farm Tree is actually a series of different applications to do different things. I'll present it very quickly now. So this is basically a tool, to promote a nature-based solution and really to deliver tree planting, at scale. And it's basically a series of applications I said before that help to document, from seed collection, going through the nursery to the planting side, and then to do the monitoring over time. And even if there is a blockchain component, to reward farmer or people who are, after tree planting, they are doing the right thing, to keep the seedling on their landscape. So we call it digitizing outcome verification and incentivize really farmer to plant trees. And I mentioned, we have a free separate application. One is called My Farm Tree Collector or My Geotree Collector. So this is basically to document seed collection, where you can take, coordinate of the trees. You can take picture. You can record the number of seed that you have collected, and the system will generate for all of each tree a specific QR codes, unique QR codes for the trees. And then once this tree also is now delivered to a particular nursery, the nursery are also registered with another application called My Farm Tree Nursery, where basically the nursery can manage, the seedling production in the nursery. So when a particular tree collected with my Geo Farm collector is delivered to the nursery can scan the code, and then it will show in the database that this particular seed load has been delivered to this particular nursery. And then seedling, are then grown in the nursery. Once they are ready to go to be planted on farm again, the planting sites are also mapped with another application called My Farm Tree or My Geo Farm. And then we know what is the provenance of the seedling planting, and then we can do the monitoring over time. And then I mentioned, the blockchain component, which is basically some kind of payment, for ecosystem services. This is basically to reward, people who are doing the right thing to keep seedling planting in the landscape, to care for them until they grow, and then can continue to grow very easily without human intervention. So we are piloting, this project in Cameroon, also Kenya, and recently we have started also to do it in Indonesia, and also in Malaysia, and also Vietnam in Asia. Just to summarize, I presented to you the work we are doing, basically to characterize, the diversity of tree species and trying really to safeguard this diversity and use them, now for different purposes. I also present a tool, diversity for restoration tool. We can really help, to select the right tree species and then for each species, where to get the best seed sources suited for this particular area. And then my farm tree, which is basically really a tool, practical tool to link all of the tree seed supply chain really to be sure that, we are delivering diversity at scale. So thank you very much. And I will have love, to be with you today, but I can't be there. And I'll be very happy to answer any question you may have by email. Thank you.
Thank you so much, Dr. Ekwe. Please give a big applause to Dr. Ekwe. Your impactful presentation has highlights the importance of conserving native species and provides essential strategies for restoring and sustaining Central African landscapes. Okay, ladies and gentlemen, dear audiences, now I would like to open the floor for questions. Please feel free to raise your hand or use the chat function to ask your questions directly to our speakers. I would like to open for probably 2 questions first. Does any audiences have any questions? Yes, we have one question here, please, the committee. Please introduce yourself and tell which speakers you'd like to have the answers from.
Okay, thank you for the opportunity. My name is Sarah Salsabil from Biology Department. My question is for Mr. Syiah Ko. So my question is there any possibilities to predict our future biodiversity using GBEF data? Thank you.
Thank you so much. So, Mr. Chijan, we have one question. Is there any possibility to predict? Is there any possibility to predict the future for using the data? However, we are here in the Azan break for 2 minutes. Maybe you can answer after 2 minutes, yeah? Thank you.
Okay.
Okay, thank you so much, Mr. Chijen. Can you please answer your question about predicting the future? Thank you.
Yeah, I mean, certainly scientists are working hard to predict how our biodiversity is going to change even the moving conditions of our environment. On GBIF, you can already find many research papers about species modeling, like using GBIF data. There are also other data sets that need to be part of it in order to produce analytical output. But I think the most important thing is we are going to do to make the future we want reality. So speaking from the perspective, I think we need to get more data available to everyone, and that's going to facilitate research, understanding of nature, and better decision about our resource management, and better informed public. And I think right now the ESG topics are heavily reliance on the result of biodiversity. So this is a great opportunity for us. According to a review at by Deloitte, €1 spent in GBIF actually can produce €12 in terms of economic output. And there are still many efforts by volunteers that are actually calculated. So this is my answer that we need to get data to put our action to work to. On one hand, we can model to see how the future may be. The direction is changing, but it's also our participation efforts that actually can make the future part of us the one that we want.
Yes, I do also believe that data is used as a tool. It helps for modeling. However, in the future, probably it cannot be guaranteed 100% if I'm not mistaken, yes?
Beg your pardon?
I think the data cannot be used 100% to predict the future, but it's only a tool for helping, yeah, for modeling.
Yes.
Yeah.
Yes. But the more comprehensive the data we have, the better quality of model.
So much, Mr. Chijan. Maybe any other participants would like to ask question? I think the presentations are very crystal. Everything has been said in 100% very completed. So without any further ado, I would like to probably give a little bit conclusion to today's presentations. The first one is the presentations from Dr. Shakir, which provides exploring innovative ways to improve soil health and biodiversity within oil palm landscapes through regenerative agriculture practices that has brought about the Industrial Revolution with UN climate action, because he, with the basis of agriculture revolution, has provided the development of industry revolution, has impacted the agriculture sector, and the impact is within the land degradation, which cause we need to think about regenerative agriculture, because this will lead to the food security, which is suitable with the Sustainable Development Goals. And then the presentation from Chijen Ko from the Global Biodiversity Information Facility Regional Asia in Taiwan, as a leading advocate for open biodiversity data, has shared with us how the use and contribution to open data through GBIF can deepen our understanding of global biodiversity, which his works underscores the value of accessible data in enabling research, conservation, and informed decision making. As well, he delve into the importance of open data in advancing our knowledge of biodiversity on a global scale and how each of us can contribute to and benefit from these data resources, for example, using R. And then the presentation from Dr. Marius has focused on identifying the most at-risk native species in Central Africa and provides critical insights for conservation efforts and sustainable landscape restoration. Dr. Ekwe's work focuses on the vulnerability of native tree species in Central Africa and aims to guide conservation strategies for landscape restoration. That he's addressed the critical need to targeted conservation efforts to support biodiversity and ecosystem resilience in the face of environmental challenges, and has specific that each species has different sensitivity to threats. Okay, maybe that's all the summary or the conclusion from the presentation that I can possibly provide today. Thank you everyone for your participation. Maybe we can give one big applause for all of the speakers today. Okay, thank you everyone for your participation and engagement in today's session. Let's continue working together to align biodiversity and climate initiatives for a sustainable future. Me, as the moderator, would like to take myself. Assalamualaikum warahmatullahi wabarakatuh.
We kindly ask the moderator to remain on stage for the presentation of mementos. The memento for keynote and invited speaker will be a certificate, and this is a certificate. Next certificate. Okay, next certificate. The memento for moderator will be given by Mrs. S.C. Harnelli, PhD. We invite Mrs. S.C. Harnelli, PhD to come to stage.
Thank you so much for our keynote speakers, invited speakers, Mrs. S.C. Harnelli, PhD, and the moderator. We kindly invite everyone to return to their respective seats. Dear guests, now we are going to watch a profile video of the Biology Department of Faculty of Mathematics and Natural Sciences, Universitas Syiah, Kuala.
and Natural Science Universitas Syiah, Kuala. Inaugurates on 17 February 1989, the Biology Department continues to develop its existence. This is proven by Unggul accreditation title that has been obtained by the Biology Department since 2023. Until now, the Biology Department has supervised 2 study programs, bachelor's and master's degrees. The Biology Department continues to be committed to developing science through education, research, and community service in order to create graduates who can compete globally.
The Department of Biology has a vision to become a center for the study of biology oriented towards tropical biodiversity in Indonesia and to provide graduates with innovative and independent socio-entrepreneurs at the global level.
In order to carry out its vision and mission, the Biology Department is supported by teaching staff that graduate from various universities in the world, with 3 professors, 7 associate professors, 18 lecturers, and 4 associate assistants. The Biology Department is qualified to develop science. The Biology Department now has several laboratories, including basic biology, ecology, microbiology, physiology, structure and development, genetics and biomolecular, and biosystematic laboratories. Lecturers and biology students are actively involved in various activities such as community service, seminars, training, and field activities that support the learning process.
The research at the Department of Biology aims to solve a number of actual problems, from ecosystem restoration to innovation in biotechnology.
We collaborate with various institutions to create synergy between science and society. Our alumni work in a variety of sectors, from research, education, and environmental conservation.
Hi everyone, I am Aigia and I am a biology alumni. Being a biology student taught me to think critically and recognize the complex connections in nature, providing a foundation essential to my growth from fieldwork to lab experience. The experiences I had shaped me in ways I never expected. The skills we developed have empowered us to make changes in research, in education, in industry, and beyond. Biology isn't just a subject, it's a way of contributing to the world.
Join us at Department of Biology, Faculty of Mathematics and Natural Sciences, Universitas Syiah, Kuala, and be part of the solutions for the future.
Have you heard this story about a beautiful place? A place that makes you feel restful. In the midst of people who live in religious harmony. A place that makes you feel home, that makes you curious for more. Adventure. Start your journey. Travel to the place where the story unfolds. Feel the sea breeze caress your face and the sound of the waves breaking your consciousness. That you can see them through the beauty of woven fabrics. Or smell them through the scent of coffee that wafts in the air. If you've never heard it before, you may now find it intriguing. You You may want to know every square inch of this place. From the beauty of its land to the depths of its seas. With an amazing biodiversity. Perhaps you've heard this story about a land which has traditions so diverse. Meet these friendly people who will welcome you with warmth. After all, welcoming guests is their tradition. The light of Aceh. Wonderful Indonesia.
Again, feel free to enjoy the snacks and drinks that have been prepared in the backside of the room. Thank you.
Thought I found a way. Thought I found a way, yeah. But you never go away. So I guess I gotta stay now. Oh, I hope someday I'll make it out of it, even if it takes all night or 100 years. Need a place to hide, but I can't find one near. Wanna feel alive outside. I can't find my fear. Isn't it lovely? All alone, heart made of glass, my mind of stone. Tear me to pieces, skin and bone. Hello, welcome home. Walking out of time, looking for a better place. Something's on my mind, always in my empty space. But I know someday I'll
Welcome back to ICBES 2024.
We have now reached the end of the event. Thank you to all participants who have attended.
And welcome back to the presenters of the ICBES 2024.
We will now announce the best presenter of the day.
Best presenter of the ICBES 2024 for online class is... Congratulations to Winati Nurhayu!
Best presenter of the ICBES 2024 for offline class is...
Congratulations to Cutnamira Putri,
Nona Rahmaida,
Wardia,
Rizka Ora Aurora Yahya,
and Tristia Rinanda.
The memento for the best presenter will be presented by Chairperson of National Work Meeting Konsorsium Biologi Indonesia, Mrs. Fauzia. We invite Mrs. Fauzia to come to the stage.
We also invite all the best presenters to the stage.
We also invite Dr. Alia Rizky and Dr. Insinyur Wiradharma to the stage.
Again, we call the best presenter of offline class. Please come to the stage to Cutnamira Putri, Nona Rahmaida, Wardia, Rizka Ora Aurora Yahya, and Tristia Rinanda. Please come to the stage.
We kindly remind to all best presenter to come to the stage.
Sekali lagi kami panggilkan kepada best presenter atas nama Cutnamira Putri, Nona Rahmaida, dan Wardia. Disilakan untuk naik ke atas panggung.
Kembali kami ingatkan kepada best presenter atas nama Cutnamira Putri, Nona Rahmaida dan Wardia untuk segera ke panggung, dan juga kami invite untuk Dr. Insinyur Wiradharma untuk dapat menyerahkan sertifikat kepada para best presenter. Kepada Dr. Insinyur Wiradharma kami mohon kesediaannya untuk ke panggung.
Thank you for all best presenters and the honored guest. Next, closing statement by the head of Biology Department of Faculty of Mathematics and Natural Sciences, Universitas Syiah, Kuala.
We invite Professor Dr. Leni Fitri to the stage.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Dear conference participant, with the conclusion of today's session of presentations, discussions, and collaborations, we have completed the first step in an important journey to strengthen cross-disciplinary collaboration in biology and environmental science. We hope that every idea that has been shared and every dialogue that has taken place during this conference will be a strong foundation for our collective efforts to face the complex environmental and health challenges of the current era. We thank all speakers, organizers, and participants for sharing their knowledge, experience, and insights. May we continue to be inspired to produce research that benefits the global community and advance our understanding of the relationships between ecosystem and human well-being. See you at the next conference. I hope next year. Keep working for the good of the environment and our common life. Thank you. Assalamualaikum warahmatullahi wabarakatuh. Special thanks dalam bahasa Indonesia kepada seluruh panitia dan peserta yang luar biasa. I love you.
Thank you to Professor Dr. Leni Fitri.
Thus conclude the first International Conference on Biology and Environmental Sciences 2024. I am Nurul Nisa Knisya.
And I am Farahdina. We are signing off. Advancing science,
preserving life.
See you at next ISEMAS. Wassalamualaikum warahmatullahi wabarakatuh.
Orang ke sini ini ya. Coba sana. Makasih, makasih. Makasih ya.